kept brought Dr.
Please.
Rupa Ghosh also. He's also in my pocket.
Sorry?
In my pocket.
Huh?
Don't give me.
It's an honor to have Dr. Rupa Ghosh as well for to be the third examiner for this competition. So, on behalf of the organizing committee, once again, I wish you all the best and let the best person win. I believe I've been told that you will have only 4 minutes of presentation. The timing is going to be very strict, so please do not cross your time limit. And then there will be 1 minute of question answers. So I'm going to invite the audience to ask questions first, and then the chairpersons and the examiners will ask questions. So please keep your questions ready for all the presentations. Okay, so all the best, and may I now request Dr. Pawan Singh to invite the first competitor.
Dr. Ananya from Institute of Pulmocare and Research.
So Ananya, all the best. Look forward to your presentation. Can you make sure that this thing is on? Yes.
Good morning, everyone. I am Dr. Ananya Botobal from Institute of Pulmocare and Research. I am working there as a physiotherapist in Pulmonary Rehabilitation Department. I am presenting a COPD case of inducted from 5th May from 2022. Dr. Parthasarathi Bhattacharya was sending this patient to me. He is the new patient for Dr. Parthasarathi Bhattacharya and me in the same day. The 70 years old male person, ex-smoker, the smoking index is 1200 and he is presenting on 5th May of 2022 with shortness of breath, cough, expectoration and chest tightness. Sir, his basic investigation is chest X-ray, PFT, FeNO, ECG and DLCO. Basics are Gold B, CAT 19, Brock scale 2, first 5 and height 160 centimeter, weight 80 kg and BMI 31.2. SpO2 and pulse rate is 93% and 98 beat per minute. Her basic blood pressure 130 by 80 and activity history 83 by FIM scale. This is the ECG, normal ECG report, and the X-ray shows here a big lungs, a big tubular lungs, a flattened diaphragm, and dilated interstitial. And the PFT report shows here the severe obstruction with 110 ml salbutamol reversibility. His symptoms is shortness of breath, cough and expectoration, and another complications is VNE. He is inducted in 5th May 2022 and the follow-ups is going on. In between the period exacerbation is also there. And in 27th July, we have conducted a peer group meeting that is one follow-up. And after the pulmonary rehabilitation, his strength in CAT score is declined, and the 6-minute walk distance in between the follow-up fluctuating first in between the period in the exacerbation period, 6-minute walk distance is not done, and lastly it is declined. And my opinion is our respiratory muscle efficiency in induction is increasing, but after exacerbations and follow-ups, it is declined. After that, it increasing.
Okay, are you done? Okay, thank you very much, Ananya. Questions from the audience? Okay, let me ask. What is the minimum change in CAT score that is considered to be clinically relevant? And what is the minimum change in the 6-minute walk distance that is considered to be clinically relevant? Because you showed some data of this one patient. Now, is this clinically significant or is this not clinically significant? The numbers seem to go high, they get better, but is this significant?
Actually, in first induction, his CAT score is 23. And after follow-ups, during the exacerbation period, CAT score is also high. And after that, lastly, his CAT score around 4.
So from 23, it came down to 4.
Yes.
That's a huge change, isn't it? What do you think was responsible for it? Only pulmonary rehab?
No,
or there were other components
no,
also?
And also other medications also. Pulmonary rehab as well as medications.
So how do you know that the effect that you showed is because of what? Is it because of pulmonary rehab? Is it because of medicines? Or is it a combination of all of these?
A combination of medications and pulmonary rehab.
So what is your main carry-home message from your presentation?
Pardon, sir, please.
What is your main carry-home message? Why did you present this case?
Actually, sir, first when I induct this patient, he's severe obstruction and severe breathlessness, and also cough and expectoration is there. So Dr. Parthasarathi Bhattacharya is sending to me to follow him up in pulmonary rehabilitation. In the first day, he couldn't walk at least for 1 minute. Then after that, in second or third day, we done the 6-minute walk test. After that, breathing pattern correction, some neck relaxation exercise, upper limb exercise and lower limb exercise, and he's also having weaning. So that's why his walking capacity is also very low. After that, he is progressing.
So this is one case.
Yes.
Majority of times, if any clinician remembers the best case with any intervention or the worst case with any intervention. So you did some intervention with the patient and you have presented it. I want to know your experience with the same intervention done on other kind of patients, other type of patients, other COPD patients. What did they experience the same kind of benefit from this intervention like this patient did?
Yes, sir. The same kind of intervention is also we almost actually we inducted almost 100,000 above patients now in above 6 months, and we categorized in 4 modules, that is module 1, module 2, module 3 and module 4. First, when we started the pulmonary rehabilitation program, we done a survey that is according to the survey, we thought that the patient is complaining the distance and time because of the some of the persons is from urban areas and some of the person in rural areas, so they did not follow continuously. So that's why we are arranging some modules. In module 1, patients are coming to our institute accordingly monthly. And this patient is stayed nearby, that is Bagoihati. He come to our institute at least weekly 2 or 3 days, and in the first almost 3 to 4 months. After that, he is coming to our institute almost 2 months around. And now he is quite okay, but exacerbation is there. Sometimes yellowish cough, expectoration is there. So then 6-minute walk distance is also fluctuating.
Abhirupa Ghosh from Bose Institute.
Some of them multiple papers. Which one?
Good morning all. I am Abhirupa Ghosh from Division of Bioinformatics, Bose Institute. I work in the lab of Dr. Sudipta Saha. Today my topic of presentation is specific sputum microbiome signatures were identified in airway diseases. So with the advent of next generation sequencing technologies, the number of studies of microbiome has increased rapidly. That also includes human microbiome. So there are many studies that give evidences of dysbiosis, that is imbalance or shift of microbiome population that is associated with diseases that includes airway diseases like asthma, COPD. And in recent years, there are few airway metagenomic studies that focuses on individual diseases of asthma, COPD, cystic fibrosis. However, we cannot compare their results because of different issues like different sample types, different sequences they use, and different techniques like 16S rRNA amplicon sequencing, where a single gene is focused, or whole metagenome sequencing, where the whole microbial genome is considered. So to address this issue, we have collected raw metagenomic data from different studies that used sputum microbiome from diseases such as asthma, bronchiectasis, cystic fibrosis, COPD, tuberculosis, and healthy smokers, and reanalyzed those raw data using a single bioinformatics pipeline. As part of the analysis, we have performed alpha and beta diversity analysis. In the first plot A, we can see clearly that healthy smoker has a diverse sputum microbiome population, and it is followed by asthma and tuberculosis sputum microbiome. And the next figure B shows that it's a beta diversity plot, and it shows that the green one COPD, the red one asthma, and the cyan one is cystic fibrosis are distinctly, the microbial population is very distinct. Next, we performed the microbial composition analysis, and we found that 5 bacterial phylum are differentially populated in different airway diseases. And we also found fungus and viral populations among the sputum microbiome. In figure B, I have shown the fungal species population, the relative abundance of the fungal species, and it's observed that Aspergillus fumigatus is relatively higher in COPD and asthma. Next, we have taken 100 most abundant species, bacterial species, and compared them, and we have got 39 shared species that are found in all of the groups that include mostly oral residents and few of the airway commensals that include Haemophilus influenza and Streptococcus pneumoniae. Along with the taxonomical analysis, we have also performed the functional analysis to obtain the pathways and genes. There are few limitations of this study, that is the number of samples that are used are very low. However, this many studies are only available, and the major problem with sputum is it contains low amount of microbial biomass, therefore the sequences contain higher amount of human reads. However, we could achieve detection of species level for bacteria, fungi as well as virus, and we could also detect the gene and pathways. So the summary is the sputum microbiome is distinct across the airway diseases, and the variation in abundance of commensals plays an important role in diseases, as well as the metabolic pathways or the species that are detected can be explored further for diagnostics and treatments. Thank you. Yes. No, we have reanalyzed the whole thing. We have only taken the DNA data from SRA. That's, yeah, only sequence data we have taken, and we have. Yeah, there are few parameters which we have to follow to like there is a protocol for quality check of the sequences. So we did that. Yeah, we have removed the poor quality sequences. And Acha, there were 5 studies that include, I can show you. These are the studies. The bioproject IDs are the studies, and their groups: asthma, bronchiectasis, COPD, and healthy smokers were included in one study, and cystic fibrosis, tuberculosis. 57. Yeah. Yeah. No, not it. Yeah. Yes. See, statistically, we found it to be overlapping. If you see the figure B, you can see the pink one is tuberculosis here, and the blue one is healthy smokers. So you can see overlapping samples. And so here, and if you see this population, the tuberculosis and healthy smokers, the firmicutes are also like almost equal amount. So it's really tough. No, it's whole metagenome sequencing. Yeah. Base calling algorithm. Yeah. Yeah, we have found artifacts, and we have removed them also. Yes, further you can culture, like whichever organisms we have obtained in a high abundance, the abundance is high, so we can just do culture, plate cultures or anything to validate them. No, so at that point what we do, the sequences which have very lower abundance, we just ignore them as abundance, as artifacts. So we only consider the sequences with a cutoff of abundance. So that's how we do. Yeah, yeah, yeah. Yeah. Yes. Definitely. That is a disadvantage of my study, at least, because we got only this much data in available databases. So this is first of its kind. Maybe later when the data increases, we can do that again. Thank you.
Good. So sorry. Some very good questions from the audience. Well done. Two of my PhD students did their PhD on this topic. So I'm surprised you did not find that in your review of literature. So Baisakhi Ghosh was one of my PhD students, and Dheeraj Agarwal. Both of them have done in asthma COPD. So COPD they have done in smoker COPD, non-smoker COPD, healthy smokers.
So whole
it's all-
genome, metagenome.
Yes, so it's all published. I think you should look that up. But I think that's a very good question that was raised. How relevant is that piece of information for an Indian population? Because a microbiome that you get from a Swedish or a Swiss population may not necessarily be
Yes, I
true
agree.
for an Indian population.
Yes, sir. So maybe we will find something different. Some population will be different, but the distinct pattern will be observed, I guess.
Okay.
One of the important things that you might notice is instead of focusing on the prevalence of the bacteria which are high in a specific population, if you focus on bacteria which are exceptionally low, especially in bronchiectasis and cystic
Yes.
fibrosis, which are almost similar to each other.
Yes, sir. In this Venn diagram, you can see the exclusive species are marked.
Yeah, can you go back to the previous slide? Yeah, this no,
This one.
back this one.
Yes, sir.
So in the subgroup C, in the chart C, if you compare bronchiectasis and cystic fibrosis, which are nearly similar diseases and both have similar morphology on clinical grounds as well as radiological grounds, you can see that Firmicutes is one of the bacteria which is normal or highly prevalent in other population groups, but it is low in these two features. So do you have anything specific about this particular bacteria? Is it good or bad, or it works against bronchiectasis or cystic fibrosis?
No, sir. I have not studied in details into it. So
So it will be interesting to find out why this bacteria is so low in these two diseases, which are almost similar on many grounds.
yes. Hmm. Yes. Thank you, sir.
Good morning. Good morning. My name is Sudipta Bhattacharjee. I am a PhD student at Department of Computer Science and Engineering at University of Kolkata. The title of my presentation is Pulmocare, a web application for spirometry-based classification of obstructive and non-obstructive pulmonary diseases using machine learning. A majority of pulmonary diseases fall into the two categories, obstructive and non-obstructive. Common examples of obstructive diseases are asthma and COPD, whereas DPLD is an example of non-obstructive disease. This obstructive versus non-obstructive classification in the early stages can prove to be an important first step in finalizing the treatment. But making an early diagnosis can be difficult due to the overlap in the symptoms of the diseases in these two categories, and often further investigations such as spirometry is consulted to make the initial diagnosis. So here we take a artificial intelligence approach in an attempt to automate this diagnostic task. So we develop a machine learning based web application named Pulmocare to classify obstructive and non-obstructive pulmonary diseases from spirometry data. To train the machine learning models, spirometry data of 1,314 patients were obtained from IPCR. This table shows the details of the patient data. The data was divided into two groups, group A and group B, where the group A data was used to train and test the prediction models, whereas the group B data was used as a blind data set for further validation. As features, the values of three metrics tested by spirometry were used. These are FVC, FEV1 and FEF. The methodology involves two components. First one is the development of the machine learning models, and second one is the development of the web application. So first, the spirometry data is divided into two groups, group A and group B, as mentioned earlier. Now the group A data is used to train machine learning models to generate a set of models which we are calling as the whole data set models. Another set of models was trained using undersampled subsets of the input data in group A. This undersampling was performed to handle the class imbalance in the input data, as this class imbalance may lead to a skewed performance of the machine learning models. After the training has been done, next the group B data is applied to the machine learning models as part of external validation. Next, the performance metrics of the modules were computed and compared, and finally the optimal model, the optimally performing model, is stored in a web server which is to be used by the web application to make the predictions. The web application mainly consists of two components. First one is the optimal machine learning model that we found, and other one is a set of scripts written in Python and PHP programming languages. Users can provide input, the spirometry data input, from the Pulmocare homepage itself. The web application receives the user request, makes the prediction using the optimal machine learning model, and displays the output as a web page again. Now Pulmocare can be accessed using this link, and it requires no installation or no registration. It is free to use. It can be used from any laptop, desktop and or mobile phone. The only requirement is that it requires a web browser and internet connection. A help page is also provided to further assist the users. This is the example of the input fields in the home page of the Pulmocare web application. The users need to provide the spirometry data input in the appropriate fields. After that, they need to click on submit to get the prediction results. Users can also provide two sets of sample data, sample inputs for demonstrative purposes using these two buttons. The output page consists of two tables. In the first table, the predicted class obstructive or non-obstructive is shown, and along with the associated probabilities for each prediction. This is shown for the 6 different classifiers trained on 6 different undersampled subsets. Finally, the total probability and the final prediction is shown in this table, and the total probability calculation is performed by averaging the probabilities of each of the classes as given by each of these classifiers. Okay. The figure 1 shows the comparison between the undersampled and the models trained on undersampled and the original data sets. So it can be seen that the models trained on undersampled data set has performed better for all of the 4 machine learning algorithms that we have performed. The multi-layer perceptron model achieved the optimal accuracy of 83.7%, and all the models show good performance in the validation data. Table 2 shows the disease-specific diagnosis of predicting asthma, COPD, and DPLD. As you can see, the optimal MLP model shows around 90% accuracy in predicting COPD as obstructive and DPLD as non-obstructive. So in summary, we developed a machine learning based web application which can prove to be an efficient and user-friendly tool for clinicians to predict lung diseases. Thank you.
Very good work. So I would like to know that this is based on spirometry only, right?
Yes.
So there are diseases like you have mentioned DPLD, which is diagnosed based on HRCT mainly. So what happens in those cases? Because we are not inputting any HRCT data over here. And for them, the spirometry data can be misleading, according to my experience. And my second question is that these are mainly checked by clinicians, pulmonologists who are already aware of the, like, what are the guidelines and what are the biomarkers, not exactly the biomarkers, like we know the FEF 2575, this is the limit, and then FEV1 by FVC, this is a limit for obstruction and non-obstruction. So is this for the non-medicals or for the medicals?
Well, for the first question, what I can say is that we have attempted to use only spirometry data, so make the prediction based on the spirometry data to see whether it is possible to make such predictions, at least statistically. And as you can see that we have achieved a reasonably good accuracy, and we have also validated the models with a separate blind data set which also performed similarly with as compared to the training data set, and there is also a little bias between as you can see the detecting DPLD as non-obstructive, it has also achieved good accuracy of around 90%, 89% to be exact. So this is the first question, and the second question is that, well, in case of so this is what we call a supervised learning approach. So in that case, the machine learning models are basically trained on some class levels which are already known, some already known knowledge. So it is quite normal to use the already known data which have been the patients who were diagnosed by some doctor, and that diagnosis is used as label to make the predictions. So this is a very common approach to in machine learning.
So one of the assumptions when it was targeting test, you have to ensure that the quality of the test is done right. So only then can you take a negative. So how confident are you that the test is not done right? Poor effort, not really acceptable, so many variables. Then we all get wrong answers because we did not go so.
Yes, so it may happen that the data might be in some way noisy, what you are saying. So statistically that may be termed as the data can be noisy. So all these machine learning algorithms has some mechanism to handle noise data. For example, the optimal model that we achieved here is the multi-layer perceptron model, which is a neural network based model. So in the neural network models, there is a parameter called a bias parameter. So upon training and upon seeing the data, the training process essentially tunes this bias parameter depending on the data set that we have. So
Okay. So one last question. Have you seen spirometry test being performed?
yes, I have seen once, but
Only once.
only once.
Okay, thank you.
Thank you.
Srijita Sen, we have got two.
Good morning, everyone. I am Srijita, a research scholar from Institute of Pulmocare and Research. So the topic of my first presentation is reversibility profile of OAD, a new look. So we know that the patients who have OAD, they have the shortness of breath due to incomplete exhalation process, which may be due to the inflammation in the airways or narrowing of the airways. So in these patients, we have checked for the responsiveness of the bronchodilators against salbutamol, which is a beta 2 agonist, and glycopyrronium, which is an anti-muscarinic agent, or AMA. So we tried to classify the OAD patients into 3 categories: asthma, ACOS, which is asthma COPD overlap, and COPD, according to their reversibility to salbutamol. And we have also tried to check for the improvement in the reversibility after giving them first salbutamol, then glycopyrronium. So in the methodology, we have recruited the patients from the OPD, and those patients who gave us the consent, we subjected those patients to the serial reversibility, first with the salbutamol and then with the GP. And the reversibility values were noted in both the absolute values and as the percentage change. On the basis of the salbutamol reversibility, we classified these OAD patients into 3 groups according to the absolute value of the reversibility and also the percentage values. And with all these data sets, we did the statistical analysis. So first we look at the demographic part, in which we can see that the COPD patients have the highest age, that is, they belong to the highest age group, whereas the asthma patients, they belong to the lowest age group, with the ACOS patient in between the two. And we can see that in the COPD and ACOS section, the male patients are more predominant than the female patients. But in case of the asthma patient, we see almost an equal number of male and females. And in case of the BMI, we can see a significant low BMI in case of COPD than the ACOS and the asthma patients. So we can see the same thing in the box and whisker plots as shown here. And next we move on to the spirometric parameters, in which we saw the FVC percentage, the FEV1 percentage, and the FEF 25-75 percentage. And we also checked for the ratio of FEV1 by FVC, which mainly show us the obstruction pattern. So here also we can see that in all the 3 cases, asthma has a significantly higher value than that of the COPD and the ACOS. We then further saw the reversibility values of both the salbutamol and glycopyrronium, and we checked for both the absolute values and also the percentage values. So here we can see that in case of the salbutamol absolute value, asthma has the highest value, then ACOS, and then COPD. But in case of glycopyrronium, we can see that ACOS has a bit of a higher value than all, followed by asthma and then COPD. But on the contrary, in case of the percentage values, we can see that ACOs have the highest value in both salbutamol percentage and GP percentage, and the COPD has the lowest value. So we can say that salbutamol reversibility is the best for asthma, followed by ACOS patient and then the COPD patients. But the glycopyrronium reversibility is best for the ACOS patient among the three groups. Lastly, I want to thank my mentor, Dr. Bhattacharya, and all my colleagues and staffs of Pulmocare and research, and all the patients who willingly participated in this study. Thank you.
Thank you. So you shot the time, huh? Questions from the audience?
Do you know what is the time of onset for glycopyrronium?
So,
Time of onset of action for glycopyrronium.
so we give like glycopyrronium, and the patient like waits there for 30 minutes, and then we check for the reversibility.
So to check for reversibility, the agents that we use, one of the most important features of those agents is that they have an early time of onset. You don't have to ask the patient to wait for such long periods. And that's why ipratropium and salbutamol has been commonly used for these things, and salbutamol has been standardized. As far as I remember, glycopyrronium is not as quick onset of, and does not have as quick onset of action as compared to salbutamol or ipratropium. But still, to use it is as a reversibility agent, you will have to ask the patient to wait for a long period of time. That's
Yes.
why it is not commonly used.
What is the clinical application of bronchodilator reversibility in these patients? How does it change treatment diagnosis?
So actually, they increase the pathway, like the what to say, like they dilates the airways, and like with the help of that, the patients can breathe out all the air, like which they were not able to do, like while because they have the shortness of breath and those things.
Good bronchodilator reversibility, not good bronchodilator reversibility. How is it useful in practice? How does a doctor use this knowledge to change diagnosis or to change treatment?
So the ones, like for example, who have good reversibility to salbutamol, they can be given salbutamol as like they can be prescribed salbutamol, and the ones who have good reversibility to glycopyrronium, they can be prescribed like glycopyrronium. The ones who don't have a like a good reversibility to GP, if they are given GP, then it won't work out properly. So I think.
Okay. Yes. So normally we use salbutamol, but you can also use salbutamol and ipratropium combination.
How many times? You will get time.
Only once, sir. Okay. It's only to test the bronchodilator.
Okay.
Only once.
Salbutamol, you cannot keep that patient on salbutamol for long. If he is asthma, you would like to use inhaled corticosteroids or LABA agents or whatever you want to choose. But to say that to test for clinical reversibility or bronchodilator reversibility and use the same bronchodilator for his symptom relief as well, that's actually not true.
Okay, sir. Thank you.
Second one.
Yes. So my second presentation is the impact of smoking on the lung function variables and reversibility trend to beta 2 agonist and AMA in the OAD patients. So here also we know that smoking is one of the main risk factor for the OAD patients, but there are some patients who are non-smokers, but they still have the shortness of breath and the related problems. So we try to see the spirometric lung function difference in between the smokers and the non-smokers OAD patients. So here we have again selected the patients randomly from the OPD, and then these patients were subjected to sequential spirometry with salbutamol first and then GP, and we recorded the reversibility according to the institutional protocol of IPCR, and we also took filled up detailed proformas in which we got to know about the smoking status of the patients, like whether they are smokers or they are non-smokers. And with all this data in hand, we did the statistical analysis part. So first we come to the frequencies of the smoker and the non-smoker in the three groups of OAD. So we can see that in the ACOS group and the asthma group, the percentage of the non-smokers are more. Here pink denotes the non-smokers and blue denotes the smoker part. So the percentage of the non-smokers are more, but in case of the COPD, there is a marginal increase in the number of smokers than the non-smokers. So next we looked at the age distribution in between the smokers and the non-smoker OAD patients. So we can see that the smokers have a higher age than that of the non-smokers, and the difference is highly significant. So next we looked at the BMI distribution, but we can see that the frequency of the BMI is almost the same in both the groups, but they have a significant difference in between the mean BMI in between the two groups. Next we again saw the spirometric parameters, which is FEV1 percentage and FEF 75 percentage. Here also the smokers have significantly low values than that of the non-smokers. We further checked for the GP and the salbutamol reversibility. So here also we can see the same trend, but in case of the GP reversibility, we can see that the difference is not so much significant. So we cannot infer anything from this data. And finally, we can say that the frequency of the smokers are more in the COPD group than the other two, and the reversibility of the salbutamol is worse in the smoker OAD than that of the non-smoker OAD, and the same trend is almost the same trend is followed in case of the GP reversibility for both the groups. Thank you.
Any questions? You looked for reversibility only in FEV1 and FEF 25 to 75. Why not FVC?
So we mainly check for the reversibility in FEV1. So because for the obstructive airway disease, we take FEV.
Okay, so maybe this could be a learning exercise for all of us who are sitting over here. In COPD, especially tobacco smoking COPD, you have small airways obstruction. So when you do a test, because the small airways are obstructed, you get a low FVC value because of air trapping. Now when you give them a bronchodilator, the air trapping reduces and your FVC value gets better. So if you have to test for reversibility in COPD patients, we should also do FVC reversibility, which is as important as the FEV1. Okay? Because FVC reversibility tells you presence of small airways obstruction, predominant small airways obstruction.
Okay.
And that's a very important parameter.
Okay, so thank you for this
So
information.
I think it's an important learning exercise for all of. You want to contribute, add anything to this?
Just a general statement. When you interpret p values, a p value which is less than 0.05 is significant. The strength of significance cannot be ascertained with the value of p. Let's say if the p value is 0.01, it is not less stronger of a relationship when compared to p 0.001. Both are just statistically significant. You cannot say that it is highly significant or it is not highly significant, it was little significant, it is just statistically significant.
Thank you, sir.
You have got the.
No, sir, two.
Priyanka Chaudhary.
A very good morning to everyone present here. I'm Priyanka Chaudhary from School of Medical Science and Technology, IIT Kharagpur. And today I'll be speaking about bioinformatics analysis for the screening of gene biomarkers in hypoxic pulmonary hypertension. So in brief, pulmonary hypertension is a condition that is defined as an elevated mean pulmonary artery pressure of more than 20 mm of Hg. And among the 5 groups of pH, my main focus is on group 3, which is associated with hypoxic lung diseases. So as indicated in my title, we are doing a bioinformatic analysis, and for that we have chosen a transcriptomics data set. So what is transcriptomics? Transcriptomics is the study of the transcriptome, which relates to the complete set of RNA transcripts that are produced by the genome under a particular condition in a specific cell. And for that, high throughput methods are used, such as microarray or RNA sequencing, which is NGS. So this brings me to how I came to the study design. The background is, as we all know, that COPD-PH, one of the most prevalent forms of hypoxic pH, leads to ventricular dysfunction and is associated with poor prognosis. And hypoxia-induced remodeling is one of the hallmarks of this disease. And though there has been a lot of studies, the underlying mechanism of the remodeling process still needs a lot of research. So it was, we envisaged that the changes in the pathways, the underlying pathways, will also be reflected in the gene expression, the transcriptome. So if one can screen the differentially expressed genes from the available data sets, we could possibly hit upon some targets or the dysregulated pathways. So in this study, we tried to identify dysregulated genes responsible for underlying molecular mechanisms in hypoxic pH, and further we validated them. So this is in brief the methodology that was used. I took a microarray data set that is available from the Open NCBI Geo database, and using a number of computational tools, we could identify 3 genes, which I'll be coming to in my next slides. So a total of 172 differentially expressed genes were identified using a computational Geo2R tool. And this volcano plot shows the downregulated and upregulated. It represents like upregulated 73 genes were found and 99 of them were downregulated. And from this supervised PLSDA model, it can be seen that these DEGs, they are able to differentiate these 2 groups, that is the hypoxic and the normoxic group. Also, we used different tools like Metascape, Cytoscape, and we come upon these enrichment analysis where we see, interestingly, chemokine-mediated signaling pathways are dysregulated, and also triglyceride metabolism and also carbohydrate metabolism was found, these pathways were found to be dysregulated. And this is the protein-protein interaction network where we see some of the most important genes that were involved. Ultimately, the 14 important genes that we narrowed down upon, the hub genes, are these 14. And we took the 3 best, the ones with the highest fold change, that is TTN, CXCL12, and CXCL9 for further analysis. So this is, in brief, the PCR, the protocol that we followed. It's the normal PCR that we do, the RT-PCR. And we took venous blood samples from COPD-PH and healthy controls. Interestingly, we did find that CXCL9 and CXCL12 were found significant. TTN was not found significant. So to summarize the study, hypoxic pH does have unique transcriptomic signatures, and the DEGs are mainly associated with chemokine-mediated signaling and metabolic pathways, as we saw from the different analysis. And two of the genes, CXCL9 and 12 in humans, did show significant dysregulation. In fact, these are associated, there are studies showing that these are associated with IL-6 related pathways, inflammation pathways, and also with macrophage polarization. So it would be interesting to go into depth and see how they are involved in these diseases. Thank you.
Yes, questions from the audience.
Hmm. That is a very good question. In fact, we are actually collecting samples to compare them with COPD as well, because as you said, COPD is also an inflammatory disease. So it would be very interesting to see whether we can compare it with COPD and see a change. So yes, I will do that. And also I'm trying to see some more markers which are related to CXL12 and 9 and get in a group of COPD as well to see how it compares with it.
I thought that was a very good question. Are you a medical?
No, sir, I am a
graduate?
master's biological sciences. So
Masters
now
in-
I'm doing PhD.
Okay, okay. So COPD patients have a lung disease that gives rise to hypoxia,
Yes,
and hypoxia gives rise to pulmonary hypertension.
yes, sir.
Uh, so pulmonary hypertension develops for a good reason, because when there is hypoxia, the pulmonary arterioles, they constrict, and they constrict because they want to maintain the ventilation perfusion matching.
Match.
Because if they don't, then you'll get dead space
Yes.
ventilation. So you get dead space perfusion. So identifying an answer for that may not necessarily give you the answer for a therapeutic thing. It may just give you an answer for better understanding the disease pathophysiology.
Yes, sir.
But that's a very good study. And as very rightly pointed out, this could be just because of COPD changes.
Yes, sir. Yes,
So
sir.
if you have COPD with no pulmonary hypertension and COPD with pulmonary hypertension, then probably you'll get a better signal.
Better, sir. Yes, sir. We'll actually
Thank
try.
you. Do you have any questions? No, thank you very much.
Thank you, sir.
Yeah, please.
Very good morning to all. I'm Sayoni Sengupta, working as a Senior Research Fellow in Institute of Pulmocare and Research. So the topic for discussion that I have chosen today is a comparative analysis of post-COVID-19 patients in the 3 waves. So as we know, COVID-19 has been with us for a certain period now. And over the recent few months, it has been reported that approximately 117 million people have been engulfed in the pandemic. We know that the infection results to a wide spectrum of symptoms with different severity. These infections may vary according to mild, moderate, or severe symptoms. So the comparative analysis in the first wave, second wave, and the Omicron phase could be an interesting comparison to analyze the exact infection status of the patients throughout. So this is a OPD-based study where we have collected post-COVID patients in the first wave, second wave, and the Omicron phase divided in a certain tenure period. The first wave comprised of 185 patients, second wave approximately 222 patients, and Omicron phase enrolled 138 patients. The patients were then evaluated on the basis of certain parameters, that is demographic profile, Borg scale for analyzing the health status, composite visual analog scale for ascertaining the cough, SOB, and functional status of the patients. And finally, two-chair test to measure the post-exercise recovery response of the patient. The comparison was done between the three waves on the basis of certain clinical parameters, comorbidity profiles, the basic hospitalization status, and the overall health status of the patients. Now, on the basis of our statistical analysis, we observed that the mean age for all the patients in the three waves were more or less the same, with the male patients being predominant in the population. The hospitalization status was reportedly higher in the second phase, with approximately 45.94% patients being hospitalized, and the duration of hospitalization was also reportedly higher in the second phase. The oxygen requirement of the patients were reported higher in the second phase again, and we can see that the overall health status of the patient was comparatively similar for both the three phases. Now, the comorbidity profile observes that hypertension, hypothyroidism, and diabetes mellitus to be the most predominant comorbidities that is reported. The spirometric factors were similar for all the three phases, and on the basis of echocardiography, conducted mainly in the first and second phase, we can see that there was a significant dipping of the reduction in the LVGLS parameter for the first and second phase. This might indicate the prevalence of myocardial injury for the patients. Now, the 2CT record shows that the post-exercise recovery responses were compromised for the first and second phase, whereas the third phase reported of less desaturation. We also observed the post-COVID symptoms or post-COVID sequels that was reported in post-second phase mainly, and saw that SOB, cough, and weakness were the prevalent post-COVID complications that were reported by the patients. So we may say in a nutshell that the analysis of second wave remained the worst corresponding to the other two waves, and the highest hospitalization and overall worst quality of life was reported in the second wave as well. So these are my references. Thank you.
Good, thank you very much.
Thank you.
If I have to ask you to point out one most important carry-home message that you would like to convey through your presentation, what is the most important change that you identified between the three waves, apart from hospitalization?
The overall clinical status, we can say, has been compromised post-COVID. I can say that post-COVID complications are the main concentrating factor that must be addressed after the pandemic.
And the post-COVID complications were very similar in all the three waves, or was it different?
No, they were similar, but certain, as I can say that the second wave indulged the most second wave indulged the most difficult post-COVID sequels. Post-second wave, I can say that the post-COVID complications were very glaring in the pandemic history or studies that have been conducted.
Is it only for number of patients?
No, the number of patients certainly plays a role in the analysis part. If I say statistical analysis, the number plays a significant role, but the overall quality of life, you can say, that has been compromised post second wave, I can say.
One of the most important factors that I think you missed out was vaccination. The impact of vaccination that changed the demographics and the severity of disease in the third wave as compared to the second wave.
That is indeed an important part, but since I have done the comparison for first, second, and third wave, so in the first wave the vaccination was not available, so I couldn't incorporate the data as such.
So, in addition to this, in the first wave, number of patients that were admitted were 48%.
Yeah.
In second wave, the number of patients that were admitted were 46%.
46%, but
the number of patients who were on oxygen were much higher in second wave as compared to first wave. The reason was that most were being just getting admitted because of the fear of the disease or
fear
for
of
the
the disease,
isolation
exactly.
purposes.
The psychological
So-
factors came into intervention post first wave, as I can say.
Any questions from the audience?
I think she has two more.
No, thank you very much.
Second one. She has the.
So this is again a continuation of the first one only. So here I try to address the active symptoms in COVID-19 patients in the three waves, the most important or prevalent symptoms that were addressed in the pandemic. So again, the most primary objective that I tried to address in this study is a comparative analysis of the active symptoms that have been experienced by the patients in the first, second, and Omicron phase in the pandemic. The common symptoms that we know were prevalent were fever, weakness, SOB, anosmia, or loss of taste. So here again, we enrolled our patients, and along with the previous mentioned parameters, we included a severity score, symptom severity score, which was calculated on the basis of severity from a scale of 0 to 5, multiplied by the duration of the symptoms that the patients said to be experiencing, and a comparative analysis for the prominent symptoms were computed. So statistically, we can see that cough, SOB, throat pain, fever, body ache, these were the prominent symptoms that we included in our study. And the first wave witnessed the highest symptom scores for SOB, body pain, and expectoration. And the second wave observed a higher prevalence for weakness, fever, anosmia, and loss of taste. We then conducted a one-way ANOVA to analyze the prevalence of the symptoms, and we can see SOB, fever, anosmia, and loss of taste were highly significant between the three waves. Fever, body ache, expectoration, and headache were mild to moderately prevalent in the three waves. So again, we can conclude that SOB, headache, anosmia, and loss of taste were reportedly the prominent active symptoms that the patients were reported with, and despite the wide symptomatic variations in the three waves, we can say that the second wave remained to be worstly reportedly quality of life sufferings. These are my references. Thank you.
Any questions from the audience?
I have just one suggestion for you. In case, most likely you are going to collect more data and present your
Yeah.
research at various different levels. Some of them will be international levels as well. Try to use as minimum acronyms like SOB or something like that.
Okay.
Use standard words like dyspnea or breathlessness. That would be much
Okay.
appropriate.
Okay.
Good, thank you very much.
Thank you.
No.
Third one.
One.
Yeah, third one. So this study focuses on the role of DSAT max in 2CT-based treatment for pulmonary hypertension. We try to develop a novel algorithm and treatment procedure for treating COPD-associated pulmonary hypertension in our clinic. The protocol for treating COPD PH was based on WHO functional status and the DSAT max for 2CT. The 2CT is a novel exercise test that mainly focuses on the post-exercise pulse rate and the desaturation that is monitored after every 10 seconds. The maximum desaturation has been used as an innovative tool to decide the treatment procedure for COPD PH. We included patients with COPD PH diagnosed on the basis of clinical radioechocardiographic criteria. The screening and selection was based on desaturation, that is DSAT max having more than 3% and a WHO classification of class 3 and 4 symptoms. The patients had received the best treatment for COPD and rehabilitation, and further with consent, the patients underwent right heart catheterization. The patients were then subjected to vasodilator treatment, that is tadalafil mainly, and a follow-up was done on a real-world protocol for ascertaining the improvement or deterioration in 2CT and CAT score. So according to our observations, we can say that the spirometric parameters were overall similar for the treatment and non-treatment group. For the treatment and non-treatment group, and we had observed a significant change in the desaturation for treatment and non-treatment group. The systolic pulmonary arterial pressure was significant amongst both the group, and similarly PVR and TPG reported to be significant. A follow-up status was observed with a mean duration of 117 days for the treatment group and 184 days for the non-treatment group, where the CAT score reported to be significant, along with the desaturation of the patients which reported to be improved in the treatment group. So we can say that the DSAT max based treatment for COPD PH showed a natural selection for the sick patients in a case of our OPD, with the PVR being reduced. The clinical treatment response of the patient appeared to be improving along with the vasodilator treatment. This is evident from our follow-up status, but of course a further investigation is recommended for innovating this in a novel algorithm for the treatment for COPD PH. Thank you.
Any questions from the audience? Yeah.
Hello. I am not medical student basically, but I am suggesting that you are doing any ROC curve or kind of thing to get a single parameter. What do you think?
See, we are trying to develop since this is a novel parameter that we are considering DSAT max to be. So we are trying to develop a absolute cutoff point, but of course with different disease population, the ROC curve may change. So that's why we are trying to develop a novel algorithm to justify that 3% is an absolute cutoff mark for the patient populations, apart from COPD, of course.
Can you please explain what DSAT actually is? Because from the data, it seems that patients who are getting treated with tadalafil are actually doing worse as compared to patients who are not getting treated. So what actually is DSAT max? How do you calculate it?
Sorry, I didn't follow.
What is DSAT max?
See, we have an innovative exercise test practiced in our institute, that is 2CT. As I mentioned here, the two-chair test, we compute a pulse rate change and a desaturation change for the patients over a period of 10 seconds. I mean, 5 minutes the test is practiced, and after every 10 seconds, we compute the change in the pulse rate and the desaturation. So the difference between rest and the maximum desaturation that the patient generated after the completion of the exercise, the difference is regarded as a DSAT max. So if the DSAT max is more than 3%, which I said we are still, exercising and brainstorming to validate it as the absolute marker, if the change is more than 3%, then we consider that as the ideal parameter or the marker for one of the parameters for recommending the treatment strategy for the patients. So that is considered as a DSAT
So if it is
max.
more negative, then it is bad for the patient?
Bad for the patient. I mean,
Can
more than 3%.
Can you go back to the next slide, please? Yes,
Yeah.
So this says that DSAT max was more negative for the treatment group as compared to the non-treatment group.
Non-treatment. I mean, -6, the -6 is considered as more negative or more severe requiring treatment strategy for
So does
the,
this mean that drug is doing bad for the patient?
this is the baseline data. This is the baseline data, and after follow-up, as you can see, the DSAT max has reduced considerably.
But it has improved much better for the non-treatment.
Non-treatment group.
Patients who are not getting treated are doing better than patients who are getting treated.
Sorry?
Patients who are not getting treated have a positive DSAT max as compared to patients who are getting treated on follow-up.
The non-treatment group suggested minus 3.37 since they were not given treatment. And the treatment group, since clinically they were manifested, so we considered treatment as an ideal step to correct their, clinical status.
So in the baseline, we are seeing that the DSAT max for the non-treatment is minus 0.3 and minus 3, I think. And for the treatment group, it's minus 6. So does this mean that homogeneous patients were not taken for this study? Because ideally, we should have equal DSAT and then we should see.
We should see, but since the treatment criteria for the patients were defined according to our algorithm, so it was obvious that the patient requiring treatment, we can say that they were more severe or more sick as compared to the patients who didn't indicate any requirement for treatment strategy.
The baseline flow is that you decided that X intervention is going to help the patient, so you gave that intervention to only those patients who were sicker.
From a clinician perspective, clinician's point of view, we narrowed a few parameters which indicated the treatment strategy or requirement apart from the ideal scenario that was presented. For example, we firstly considered the clinical status of the patient defined by the quality of life. Then the echocardiography played a significant role for deciding whether vasodilator treatment should be indicated or not. And finally, we considered DSAT max also as a parameter to help us drive the treatment strategy.
Done, I think, all three.
Sanjukta Dasgupta from IIT Kharagpur.
Good morning, everyone. Myself, Sanjukta Dasgupta from IIT Kharagpur. The topic of my discussion today is meta-analysis of mortality-related risk factors in hypersensitivity pneumonitis. So, hypersensitivity pneumonitis, as you all know, that HP is a type of DPLD that happens due to inhalation of certain antigens. This antigen may cause inflammation into the lung parenchyma, which can slowly progress into pulmonary fibrosis. These are the common symptoms that are associated with the disease. If we see the prevalence of the disease, it varies from country to country depending on the geographical location, environmental factors, etc. Interestingly, in Indian scenario, according to ILD India registry, it is one of the most common type of DPLD. Another interesting fact associated with the disease is that the death rate associated with the disease has been increased very significantly in recent years. Keeping this thing in mind, there are many population-based studies that has been performed all over the world to identify the mortality-related risk factor of the disease. However, there are no cumulative study that can combine all these risk factors and come up with the conclusion that which risk factors are the most significantly associated with the disease mortality. Here we will be doing a meta-analysis. So what is meta-analysis? Meta-analysis basically systematically combine all the previous research articles that are focusing on a particular topic. Here this particular topic is the mortality of HP. So after combining all these articles together, we will come up with the conclusion that which risk factors are the most significantly associated with disease mortality among all these cofactors. So this is the overall work plan of our study. here using these keywords, we have searched these databases, PubMed, Embase and Cochrane Library, and we have come up in the 22 relevant study. So these are the performed all over the world, which are precisely focusing on the mortality of HP. Then this is the country-wide subdivision of these studies, and then we seen the strength and association between the mortality and the risk factors using the odds ratio. This is the baseline characteristics of these studies, these 22 studies. So now coming to the odds ratio. So odds ratio basically quantifies the relationship between two events. So here event A is basically these common risk factors that are present to be associated with the disease mortality among all these 22 studies. And this event B is the mortality of this patient. This odds ratio value actually varies. So if this value is more than 1, that means there is some association present. And the more the odds ratio value means higher the association between these two events. So now coming to the result of our study. As you can see here that advanced age male patients and the patients who are having honeycombing and traction bronchiectasis have the highest odds ratio among these all 11 factors. Also, if you can see the p-value, these values are significantly associated with disease mortality. Furthermore, we have done a subgroup analysis. We have taken only the chronic HP patient from this HP patient pool. There are total number of chronic HP patient here are 720. And when we did the same type of analysis to identify the mortality related risk factors, we have seen the similar type of trend here also as the HP. So from this study, we can conclude that elder patient, male patient, and the patients who are having extensive fibrosis, that is honeycombing and traction bronchiectasis, are associated with higher risk factors. Also, we need to do some multicentric cohort study to validate this finding globally. Also, if we can find out some optimal cutoff values for these HRCT patterns, it can help the clinicians to identify the patient who are at higher risk of mortality. These are my references. A part of this work has been published in Expert Review of Respiratory Medicine. Thank you.
Good, thank you. Any questions?
Yeah. So no work has been done from India, like what are the risk factors for the Indian population?
Till now, no population-based, but there are some studies from Asian population, from China and Japan, as you can see.
And is there a difference between the Asian population and say the Caucasian population in terms of the risk factors for mortality, or they are similar?
That's a very good question, but here we are trying to see the global risk factors. That means the risk factors that are associated with HP throughout. That's why we are doing a meta-analysis, not a population-based study. So this is the factor we are considering here.
Can you go to the results?
Yes, sir.
Very short, the p-value here. So why did you miss out lower FVC, lower DLCO, ground glass opacity? They were also statistically significant.
Yes, sir. Here we are seeing the odds ratio as well. So odds ratio actually gives the relationship between the presence of these patterns and the mortality. So since it is the highest presence, based on the odds ratio, we are selecting the patterns.
But so lower DLCO was associated with higher mortality?
Yes, sir. But this is statistically significant. Lower DLCO is statistically significantly associated, but the if the odds ratio value is less than 1, that means there is no association. This association is, if the association is not higher than 1, that means it is not associated.
Just to clarify, the number 4 in advanced stage means, let's say roughly, or for layman, it is 4 times increasing the risk of death.
Yes, sir. Yes.
So with this chart, you mean to say if the patient has low DLCO, he has low chances of dying. Because
Sir,
the ratio is less than 1.
Yes, sir, these are the all 11 factors we have screened from the 22 studies. So all of these are associated. All of these have been associated in some population-based study. That's why we have screened this. But when we see the global picture to see which one of the highest odds ratio, that give us the idea that elder patient, male patient, and these patients are having extensive fibrosis are associated with higher mortality risk. This is a global picture of our study. This is basically lower FVC is,
What is likely to have happened is, it's not lower FVC, it is FVC.
yes,
FVC, and not lower DLC or DLC. So if the FVC is on the lower side, then you get a higher risk of dying. But the risk is, the odds is quite small. Nonetheless, but, I think it's just a error in the mentioning of it. Any more questions?
thank you, sir.
Devraj Roy.
I don't think so she is in any.
Good morning to all. I am Devraj Roy, working under the guidance of Dr. Raut Banerjee and Partharthi Bhattacharya as a PhD student. So my topic of presentation is established fibrotic versus potential fibrotic DPLD as spirometric appraisal. So when we look at the global mortality of DPLD, it is increasing. In India, the death rate increased from 2.3 to 4.1 per 1 lakh people. And with this increasing disease, there is a need of diagnosis of DPLD. We all know there are many diagnoses of DPLD. HRCT is a very prominent one, but the imaging analysis or serological test or the other analysis that we have, that is quite expensive, and they need a certain amount of infrastructure, which is not very present in rural India. But pulmonary function test, which is present in many clinics, and pulmonary function test can be done in a portable disease, which is also non-expensive. So first we focus on FEF 2575, because in previous work from our lab we have studied that FEF 2575 can distinguish DPLD, and here the sensitivity is very good and the specificity also is very good here. So our observation is that FEF 2575 is higher in fibrotic patients, and with this observation we are going to the methods, and we take 177 of DPLD patient, and we are based on HRCT, and honeycomb and traction bronchitis, it is a two factor that is associated with the fibrotic, and we take sharp reticulation as a characteristic of potential fibrotic, and then we take the spirometric parameters, and then we go for the statistical analysis. So in the result we can see FEF 2575 is not significant here, but there is a trend that FEF 2575 is more in case of established fibrotic patients. Then we work with the derivative parameters of spirometry, and here we can observe that FVC by FEF 2575 is very significant with the percentage and absolute value also. So when we does the other principal and component analysis and differential analysis, so we get a VIP score maximum with this ratio of FVC by FVF, and in the T-score chart we can observe a strong overlapping region. This is quite natural because we are analyzing a same disease, which is DPLD, but there are some point which is far away from this overlapping region, and we can focus on these points as a diagnosis of fibrotic DPLD. So what outcome of this experiment, that in case of fibrotic DPLD, fibrosis in the outer layer can pull the small airways. So that's why the small airways are failed to collapse, and that's why the FEF 2575 has increased in this fibrotic DPLD, and we can use this FEF 2575 as a diagnosis tool after modeling. Actually this is a pilot study. We are planning to do develop a model on basis of FEF 2575. Thank you.
Showing a significant p-value is one thing, and then showing the strength of that through sensitivity, specificity, positive predicted value, negative predicted value. I think that would be more important.
Yes, that is very important. No, no, no. That's just a pilot study. We have observed this only, and after taking more patients, we want to increase the patient number, and then we go for a this kind of test, is ROC curve and this differential test.
Okay, I think that would be more important. But
Yes, yes.
but so you're saying that you're getting a signal, but a signal from FVC divided by
Yes, FVC divided by FF, 25-75% is an absolute value. There is a strong signal there.
and you don't have a healthy population in this.
Yes.
Do you have a healthy population in this?
No.
No. So we do not know what would be the values in a healthy population.
Actually, we have previously done a study in our lab that FF 25-75 can differentiate the obstruction and DPLD, obstruction and restriction with a very high percentage of significance. Actually, that's why we didn't take the healthy population this time, but we will include in your.
I think you should, because that will give you a stronger signal.
Can you please tell me how did you classify the two types of fibrosis?
Exactly, based on HRCT actually. When we, it is done by Partharsarathy Bhattacharya, sir, when he observed the honeycomb traction bronchitis is there, we group them as a established fibrotic, and when his sharp reticulation is present, we termed as them as a potential fibrotic patients. This is the two group that we have. Okay, thank you.
Thank you. Shubham Ghosh.
So we also have Dr. Unithan with us. Sorry that she came a little, but maybe she was sitting behind. We didn't know that.
I actually came half an hour ago.
Oh, I'm so sorry. We did not realize that. Very sincere apologies, but so she's also going to be one of the examiners for you. Yes, Dr. Shubham.
Good morning, sir. Good morning, everyone. Today, I'm Shubham Ghosh. Today, I would like to talk about spirometric comparison between the Th2 and non-Th2 subtypes of OAD population. So, OAD population can be categorized into two subtypes based on the T helper type 2 cells, that is Th2 and non-Th2. So, here we categorize Th2 and non-Th2 subtypes. The Th2 non-Th2 further classified into absolute Th2 and possible Th2. These were done based on the values of FeNO, absolute eosinophil count, and the IgE values. So, although in most of the cases Th2 can be defined as high eosinophilia, but this is not the case. In some cases, FeNO and IgE also play a role to define type 2 Th cells or non-Th2 cells driven asthma. So our objective was to look for the distribution of Th2 and non-Th2 in the overall OAD population and to go for their spirometric and demographic comparison. So we randomly collected patients from our OPD, and first we did a spirometry to classify them as asthma or COPD, and also we did FeNO, IgE and AEC and categorized them in absolute Th2 or possible Th2 and or possible non-Th2 and absolute non-Th2. So from the overall OAD population of 240, we found that asthma got a little percentage than COPD, and when we go for Th2 and non-Th2, we found that Th2 population is prevalence in the OAD population. We further classified asthma and COPD in Th2, non-Th2, and we also look at the percentage of absolute Th2 and possible Th2, and also absolute Th2, absolute non-Th2 and possible non-Th2. So these were the percentage values in the population and the frequency of distribution. So when we compared the Th2 and non-Th2 in overall OAD, we found there are significant differences in demographics and in spirometry, especially in FEV1, FVC, FEF20% and the reversibility parameters. We further go for asthma possible TH2 and compare with COPD possible TH2, and same we did for asthma possible non-TH2 and COPD possible non-TH2, further absolute asthma TH2, COPD absolute TH2, and also for asthma possible TH2, asthma possible non-TH2 and COPD possible non-TH2. So if we see the overall chart, there are certain significant differences between most of the parameters. So in conclusion, I would like to say that TH2 and non-TH2 phenotypes differ significantly in the OAD population, and further classification of asthma and COPD into non-TH2 and TH2 have several significant differences. Therefore, the TH2 based classification may help us to understand the etiopathological development and can guide the therapeutic approach in a better way. Lastly, I would like to thank my mentor and guide, Dr. Parthasarathi Bhattacharya, and I would like to thank all the colleagues and staff of IPCR. These are the references I have used for. Thank you.
So are you trying to say that in COPD patients, you want to see the subtypes of TH2 and non-TH2?
Yes, sir.
Okay. And how did you distinguish these patients?
I have classified TH2 and non-TH2 in these three parameters, FeNO, AEC and IG.
How reliable is this?
So I have used a reference actually. Most of the papers say eosinophilia is the only parameter, but there are some of papers they can say FeNO and IG can be used for substitute. So for absolute TH2 we took all of the parameters high.
All three parameters.
All three parameters high, and for possible TH2 we took only any of the two parameters.
Yeah.
Exactly.
Right, sir. Yeah, that's right. Actually, I have compared the whole TH non-TH2, but non-asthma TH2. And
Are you trying to identify the ACO variety of COPD?
I have not included ACOS in this study.
But you must have, because the TH2 type of COPD, that means it is echo.
Echo, yes.
Good, thank you.
Thank you, sir.
Rick Chakraborty. You have got two. Yeah.
Good morning, everyone. So may I?
Yes, yes.
So today I'll be reporting like factors affecting the feasibility of subjects to attend a COPD rehabilitation program in the real world and appraisal. Means, first of all, we have initiated a peer program in our institute, and we have checked for the feasibility of the patients to join the program. We have checked for the feasibility of the patients to join the program. Since there are patients, the first problem that we face in patients, like they are not all aware about the peer program and all these things. So what are the main obstacles in the patients joining a peer program? That was our interest to find out what are the major obstacles for the patients to join the peer program, and it's on both the sides, as on our side as well as on the patient side. So we conducted a Google survey with a few questions set on consensus by the team, and for that we wanted to check what was the feasibility. For standard pulmonary rehabilitation, patients need to visit twice or thrice a week for around 6 weeks to have enough changes or enough expected outcome. So that was the motive. So when we formed the questions, we formed on patients' degree of interest in joining a peer program, feasibility of comfort of visit frequency, as well as the major perceived obstacles for them to visit the peer program. And every question was scaled on a 0 to 5 basis, where 0 meant they have no interest, where 5 was like the maximum possible value. What we found out that almost all of the patients, around 95% of the patients were interested in joining a free pulmonary rehabilitation program. And the patient cohort, as we have seen in our case, we had 200 patients. Of them, the male-female ratio was around 67 to 30%, on a roughly basis 70-30 ratio. And once we checked about the feasibility for their frequency of visits in a peer program at a stationed institution, it was like the highest was once every 12 weeks. That is in 3 months visit, followed by monthly visit, which was then followed by weekly visits. So the basic obstacles that we found out in a patient's joining a station peer program was distance and location in this case, because there are patients who come from very long distances and travel long distances. So for them, it's not very much possible to join a peer program on a daily basis. So that was the problem that we found. But one thing was there also in our patient, in this cohort, the patient's average age was around 60 years above. So for them, the main problem was their physical disabilities in some cases, as well as lack of attendance, which we face very much in today's life. So what I told just now, I have just shown this in the next slide. So this was the frequencies, and this is the dominance of the male and all these things. So basically, whenever we are concerned about patients visiting a peer program, these major obstacles are coming into action. But if we change the cohort, we change the number of the patients, this might vary, but this was a pilot survey and this is our inference. Thank you.
So what proportion of your baseline COPD patients were male and what proportion were females?
Sir, almost all of them were COPD patients with moderate to severe means obstruction, sir.
No. So in your COPD population,
Yes, sir.
how many were males and how many were females?
Sir, the ratio was 67 to 30, means 70% were male and 30% were females,
Go
sir.
back to the previous one. So
Yes,
no, the next one.
This one?
There was male predominance.
Yes, sir. Around 70%, sir.
So this is the patients who took part in the survey, or this was a baseline?
No, sir. This was a survey, sir. This was a OPD survey, sir. Whichever patient was there who were willing to take part in the survey, we assessed them, and the criteria was they were all having COPD.
So what percentage of the males said yes? What percentage of the females
Okay.
said yes?
That I need to show, sir. I haven't shown over here, sir. That I need to show. So mostly all of them were interested in the program, but since this is a new thing for many of us and it has come into action post COVID era, so they are well interested and all of them are interested, but there is certain reluctances females sometimes, but males are okay, we'll join the program, but we can't come. So if there is any possibility of any remote sessions or all these things, then possibly we can join. So we're working on that.
So, for all the research, the main purpose to do a research is to increase the generalizability or the applicability of particular thing.
Yes, sir.
That's why everybody does research, and
Yes, sir.
to how that we can change our practice. So I'd like to congratulate you, because this is the most practical study that we have seen today, which actually tells us some kind of reasons the patient is not able to attend the rehabilitation programs. This is one thing. Second thing, most important part to do a survey based study is, what is the most important part? What do you think?
Sir, basically what we started our study in this case, we are setting up a pulmonary rehabilitation program at our
What is
center.
the most important part?
Yes,
Do
I'm
a
coming.
survey-based study.
Yes, sir,
Simply a
it's
survey-based
the
study.
means interaction as well as the means what I should say. It should be the homogeneity of the data.
Most important part to do a survey-based study is the questions, because it is easy to implement. You can send a Google link and they will fill the form on their own,
Yeah.
whatever. So it is easy to get responses from people, but if you ask the wrong questions, then the responses will be irrelevant.
You don't, yeah.
So if you had added some more questions in this study, like is there a feasibility to do a virtual rehabilitation program for you? Would you be able to participate in that? If yes, then what kind of platform would you like?
Yes, I'm coming to that, sir. Sir, actually, this was an initiation we have, means pilot survey we had taken, so that we can initiate the protocols in which we will be categorizing the patients to join the peer program. So that according to that, we have modulated 2 modules, like who can come frequently, who can come less frequently, and then there is another group who won't be able to come regularly. So that we have seen over here. If I can go to the next this slide, sir, who can come over in 12 weeks, that is at least at a 3 months interval. For them, we have at least initiated remote monitoring sessions, like on video call as well as on WhatsApp, as well as on group meeting sessions, so that we can monitor the patients for like whatever they are developing, their exacerbation types and all these things. Okay. This we have developed. Sir? Yeah, I have got one more presentation, sir. So in my next slide, I will be speaking on relation of peak of flow with spirometry variables. So this was a question that came to our mind, that in there are cases like when we surveyed the patients, we saw that there were patients with COPD who were coming from very long distances and probably rural sides, where probably there are not chances of getting one spirometry done. So we're looking for if there is any correlation between simple spirometric results where we can use that result as a surrogate for a machine-based spirometry. So we looked into the peak curve flow and its correlation with the spirometric variables, where we studied a cohort of 38 COPD patients and studied the correlation of PCF with FVC, FEV1 and other spirometric variables along with 6-minute walk distance and CAT score. The findings are as follows. Like we have got highest correlation of peak curve flow with maximum voluntary ventilation, which is also a representation of muscular efficiency. So we have got 2 correlations, we have got correlations, but the better correlations are FEV1 as well as MVV in this case. When we have seen the correlations and the significance, these are the values we can see over here. So we think the PCF can be a workable surrogate in case of surrogate of MVV in resource poor situation, but this needs to be needs more investigation in the part, sir. It's just a report we have just studied, and that's for it, sir. Sir, with a peak flow meter, sir. Cough, sir, it was peak cough flow, so cough it, sir. Cough, yes, sir. And we recorded the maximum volume, sir. It was a three times trial, and on the average, which one was the highest, we took that data, and it was done by a PFT technician, sir. Means a technician.
Normal values of any, or have you taken any control subjects?
No, we didn't have any control over that time. It was
So
only the recorded
which
data.
value did you think would be abnormal?
Ma'am, most of them were COPD patients. In this case, we haven't recorded the normal values over here. It was just to check whether this was having any correlation or not. We'll definitely look into that matter, and since it needs further investigation in that part, we're yet to complete that. Sir? Sir, both are required, sir, because if we are not having any normal value, then we can't say whether the patient is transitioning to a COPD part. So for that, yes, sir, we need to normalize the value, sir, and we are working on that.
I think more than the neuromuscular function or dysfunction,
yes, sir,
critical variable to determine peak cough flow will be your airways obstruction.
Sir?
So if you have small airways obstruction,
yes, sir,
then the force that will be generated will be weak. Or if you have a very distal lung disease like say pulmonary fibrosis or something, then the cough flow will be very weak. And therefore it's just not that, but maybe different severities of COPD will give you different cough.
Sir, this is what we're thinking of. Like we have first correlated this data with the spirometry variables. Next, we're thinking of correlating and looking into the data with the FOT variables as well. So in FOT, we can see like if there are any obstructions or restrictions in the smaller airways, that can be portrayed as well. So if there is any correlation, we can find out. Probably that will work out. That's my view, sir. Actually.
Okay. Okay. Fine. Please,
Thank you,
please,
sir.
please.
Yes, sir. Sir, probably I will need to look at that, sir. Probably I will need to look at that, but I'm not sure. I haven't checked it. Sir, yeah, Pearson's are, sir. Actually, yeah, that is true. That is true. Okay, so that I will need to look on once again. So there is no, at least what I have found out, there was no skew, but skew in the graph. Sir, I will definitely look into that. Okay, I will look into that. I'll definitely look into that, sir. It's a pilot study, sir. I will definitely look into that. Thank you, sir.
Good. Thank you very much. Thank you. So, any concluding? No, so thank you very much for all those who participated in this competition. I think all the presentations were very good, excellent. Some of them were top class, some of them were good. The research questions that many of you all asked were very good. So I think I must compliment all of you all for putting together such wonderful research for the presentation here. More important than that, I like the quality of the questions that came from the audience. I think that was also very good. So all the questions that you asked, I think gives a different value to this kind of a competition. So thank you very much. And on behalf of all the other co-chairs, I would like to thank each and everyone, and especially the organizing committee for hosting such a wonderful competition for the Pulmocon 2022. Thank you very much.
This concludes our poster session. The next session is Phenotyping of Obstructive Airway Diseases, COPD, its Applied Importance. The speaker of this session is Dr. Debashish Behra. To chair the session, I would like to call Dr. Saurav Majhi on the dais, please. Dr. Majhi is a dynamic pulmonologist trained from premier medical institutes. He has done his MBBS from Calcutta University and then from PGI MER, Chandigarh. He has special interest in managing complex asthma and lung cancer patients. Sir, I welcome you.
The second session of this hour is phenotype of obstructive airway disease, COPD and its applied importance. The speaker is Dr. Devashish Behra. He is a MBBS, MD Pulmonary Medicine and DM and DNB. He is very severe academic. He is an academician and several publications to his credit. Presently, he is attached as an assistant professor in pulmonary medicine in Kalinga Institute of Medical Science, Bhubaneswar, Odisha. Dr. Devashish Behra, please.
Thank you, sir, for this nice introduction. Good morning, everyone. First, I would like to thank organizing committee for providing me this opportunity. So I have been allotted the task that is phenotyping in COPD. So it's a pre-T session probably, so I'll be not taking much of your time. So within next 15 minutes, I'll be mainly focusing on how to treat your COPD patients based upon your phenotyping. So before going into detail of the topic, let me ask a simple question to all of you. How many of you generally write in your prescription while seeing a patient of COPD in which stage the patient is and what phenotype does the patient belong to? Anyone in practically? Sorry, ma'am? Most of it. Thank you, ma'am. So going to the topic, so we all know that COPD is a complex and heterogeneous disease. So what do I mean by complex? That means COPD has a number of intrapulmonary and extrapulmonary components whose dynamic interactions along time are not linear. And coming to heterogeneous, it indicates that not all these components are present in all the individuals at any given time point. This realization inevitably leads to the need for personalizing the assessment and treatment of patients with COPD. Our understanding of COPD has changed dramatically over the past 2 decades. We moved from an airflow limitation, that is FEV1 centric view of the disease, to the realization that COPD is a complex and heterogeneous condition. So what is complexity? If you see the first figure, this is a heterogeneous form. You can see all the subtypes of COPD in the first figure, indicating in the color like blue, red, like that. And in the second figure, you can see the COPD phenotypes, the subgroups, the blue in one part, the green in another part, but the red in third part. But the practical scenario, if you will see the group C, we often see most of the phenotypes often intermingling with each other. We might not see a single phenotype while treating a patient of COPD. We might see 2 to 3 phenotypes of COPD existing in a single patient. So what is the definition of phenotype? The definition for phenotype is considered as the physical appearance and biochemical characteristics as a result of an interaction between genotype and environment. And moreover, the definition clearly states that a phenotype has to be a subgroup with a great impact in the progress in the form of symptoms, exacerbation, response to therapy, rate of disease progression or death. Coming to COPD phenotypes, the classical COPD phenotypes way back we all know, that is chronic bronchitis and emphysematous type, and some new COPD phenotypes that are upcoming nowadays. Classical COPD phenotypes, as I have already mentioned, this chronic bronchitis and emphysematous type. And coming to the limitation of the current therapy in management of COPD, if you'll see, there is lack of scientifically robust studies clustering COPD patients according to the phenotypes. Detailed analysis of the various clinical characteristics phenotypes of the patient is missing in the gold document, and recommendation of modes of treatment based on phenotyping of patients of COPD are at their infancy. Then why COPD phenotypic guided treatment is important? Because of significant heterogeneity of clinical presentation of COPD. Not all kind of treatments are suitable for all the COPD patients. Phenotyping or subgrouping is essential to tailor the management of an individual patient with COPD and to predict who will respond to the specific class of drugs, leading to personalized treatment. So this is the recommendation of phenotyping in management of COPD in the gold document. In 2019, if you'll see, gold guidelines management for stable COPD was redefined. Group A, B, C, D are now just used for informing the initial treatment only depending upon the MMRC and CAT score and the history of exacerbation. And regarding follow-up, if you'll see, two outcomes are proposed, that is dyspnea and exacerbation with different individualized treatment algorithm. And blood eosinophil count is introduced as a biomarker for the likelihood to treatment with an inhaled corticosteroids. If more than 300 we can use, or 100 to 300 history of exacerbation we can use, and if it is less than 100 of eosinophil count, it's better to avoid the risk of pneumonia. Then this is the widely accepted COPD phenotypes in many papers. You can see a lot of varieties are there, starting from chronic bronchitis type, emphysematous type, asthma COPD overlap type, frequent exacerbator when the number of exacerbation is more than equal to 2 per year, and rare exacerbator when it is there is no exacerbation or just one exacerbation. So this is the most widely accepted phenotypes in most of the papers. Some emerging COPD phenotypes are there, like COPD with bronchiectasis when there is evidence of bronchiectasis in HRCT. We all know that if you will do CT in a COPD patient, around 10 to 15%, 50% of the patients might show evidence of bronchiectasis in COPD. Then upper lobe predominant emphysematous type. As the name suggests, there is a predominant emphysema in the right upper lobe. The fast decliner type, when there is rapid decline in lung function. Then the combination, the comorbidities of systemic phenotype type, alpha-1 antitrypsin deficiency type, non-smoking or non-smoker COPD type, and a very rare variety, that is pulmonary cachexia type, when the BMI is less than 21. So coming to the phenotypic guided COPD care, if you will see, first coming to asthma COPD overlap. So what is the clinical importance of this phenotype? You will see there is increased frequency and severity of exacerbation, faster FEV1 decline, increased healthcare utilization, and increased comorbidity, burden and mortality. Identification, I am not going, you all know how to diagnose this echo. And coming to the management, if you will see, the inhaled corticosteroids form the main stem of management in those variety of COPD phenotype, and you should always assess the atopy. If the patient is having concomitant atopy, you should address it and treat it if the patient is not responding to your conventional treatment. Then coming to frequent exacerbator phenotype, there is faster decline in lung function, increased healthcare utilization, increased mortality up to 3-fold, and increased risk of depressive symptoms. Definition I have already mentioned, when there is more than 2 exacerbation per year, it is known as a frequent exacerbator. And coming to treatment, long-acting bronchodilator with inhaled corticosteroids form the main stem of management, and you can also add the anti-inflammatory treatment like macrolides, roflumilast on N-acetylcysteine in managing those kind of patients. Then coming to upper lobe predominant emphysematous phenotype, the main clinical importance is that those kind of patients are typical candidate for lung volume reduction surgery. They do very well after the lung volume reduction surgery. Then rapid decliner phenotype, the clinical importance is that this group of patients have a very high mortality, and you can identify those group of subtypes by rapid decline in lung function. They are relatively younger, poor nutritional status with no major comorbidities. And the treatment important is that you can send those kind of patients for early initiation of lung transplantation. Then coming to comorbid phenotype, there is increased healthcare utilization, poor quality of life, and increased mortality risk. And you can identify those group of patients with persistent symptoms despite comprehensive therapy, and the symptoms are disproportionate to the severity of airflow obstruction and high comorbidity burden. And management, you have to address the comorbidities apart from your conventional management for COPD. Then this true, that is physical frailty and emotional frailty type. This clinical importance being is that high symptoms burden, frequent exacerbation, poor functional capacity, and poor disease management in both these phenotypes. And you have to manage those group of patients with pulmonary rehabilitation for physical frailty type, and behavioral intervention and pharmacological treatment for this emotional frailty type. Then several papers are there, they have classified the COPD phenotypes for the ease of this, like one paper is there, they have classified the COPD phenotypes into 4 categories, that is non-exacerbator, asthma COPD overlap, emphysema exacerbator and chronic bronchitis exacerbator. Basically, the difference is in the management, how you will manage those kind of patients. In non-exacerbator, usually one or two bronchodilator is sufficient. In asthma COPD overlap, you have to add inhalational corticosteroids, biologics, and bronchial thermoplasty in a couple of cases you can consider. In emphysematous type, you have to add dual bronchodilators, mucolytics, and you can consider the patient from lung volume reduction surgery. And chronic bronchitis type, you have to add microlides therapy, mucolytics, and mucus clearance in those kind of patients. Similar, another paper they have described these 7 types of phenotypes. So I am not going into the treatment. It is basically same as I have mentioned in the previous slide. This is a very good paper of the Indian population. They have taken 100 patients of COPD to identify various phenotypes of COPD, clinical assessment, functional assessment, spirometry, and radiological assessment also. And what they have seen, you can see, out of 100 patients, 45 patients belong to this emphysematous group. Then asthma COPD overlap and chronic bronchitis constitutes around 20% each, and 15% belongs to COPD with bronchiectasis. And if you see the board index, most of the patients belong to second quartile of the board index. Coming to the modified MMRC scale, you can see most of the patients were belong to grade 1 of MMRC scale, followed by grade 2, then grade 3, then grade 1. Then this question often asked in our day-to-day practice, that which stage of COPD patient you usually encounter in your day-to-day practice while seeing a patients of COPD. You can see the stage 3 is being the most common phenotype, most common stage we usually see in our OPD practice, followed by stage 4, then stage 2. One must go through this article. This is the Indian population article. They have very beautifully described what is the phenotype, what are the stages of COPD, what is the MMRC grading in different phenotypes. Then not only at the initial evaluation, you should divide your patient what kind of phenotype he or she belongs to. During also exacerbation, a couple of authors have described the phenotype depending upon your exacerbation. This is the most important paper that I have found. They have described the exacerbation as per the phenotypically type A, B, C, D, E, F, G, X. A for airway infection by virus. You can see when the viral PCR is positive in exacerbation patient, then it is known as A type. B is for bacterial infection, that is evidence of bacteria in the sputum culture, and also CRP more than 20. C is co-infection when both viral and bacterial infection occur. D is sometimes patient admitted because of recurrent depression and anxiety. So this category belongs to category D. E is of eosinophilic variety. F is failure, that will be cardiac failure. We can diagnose cardiac failure by sending NT-proBNP or high sensitive troponin I. And sometimes G is for genetic general environment, when there is change of environment of a patient from warm to cold. Sometimes there is exacerbation of COPD. So that phenotype is known as general environment. And X is obviously when the etiology is not known. And if you look at into the data, virus were detected in around 30% of the patients. And those patients in which virus were detected, those patients had longer hospitalization with lower mortality. The hospitalization was longer in those kind of patients, but the mortality was lower. Bacterial infection was found in around 40% of the patients and is mainly seen in frequent exacerbator group. And this eosinophilic exacerbation was associated with lower pH, higher PCO2, greater use of non-invasive ventilation, but shorter hospitalization and lower infection rate. And cardiac dysfunction, severe anxiety, depression were common both in infective and non-infective exacerbation. This you can see the survival at 12 months post-discharge based upon the presence or absence of infection in acute exacerbation of COPD. As I have already mentioned, when those patients who have viral infection, the mortality, the survival was much better as compared to bacterial infection or when no etiology is found. So what is the impact of phenotyping in the management of COPD? It has changed the non-pharmacological and pharmacological management of COPD. Common reasons for the multiple failures of drug development in COPD have been described and analyzed, like including inadequate target engagement of the drug, poor patient selection, and use of wrong clinical endpoints. And use of biomarkers for COPD phenotype patient dramatically increases the probability of success of novel drugs in phase 2A trial from 29% to 82%. So to conclude, COPD is now widely accepted as a heterogeneous condition with multiple phenotypes and endotypes. Phenotypical approach to COPD is having huge impact on everyday practice. Phenotypical approach is a small step to precision medicine in COPD management, and it is suitable for more personalization of the treatment of COPD. Thank you all for your patient hearing. If any question is there, I am happy to answer. If
Thank
any
you.
suggestion is there, my teacher Saibal Mitra sir is there. He can give any suggestion.
Thank you, Dr. Devasis, for your nice presentation. Can I have one question?
Yes, sir.
Is there any difference of the phenotype in male and female COPD?
Male and female COPD.
COPD. Is there any, which one is more in female COPD?
Sir, in COPD, if we compare between male and female, those patients who present with male phenotype of COPD, BMI is usually low, sir. And as the male COPD are mostly smokers and females are non-smokers, so obviously the difference will be there in terms of cardiovascular morbidity or maybe chance of pneumonitis like that.
No, or whether we are getting the more echo or more eosinophilic in case of the female. Is there any evidence?
No, sir. I have searched my literature. I have not found any evidence regarding.
Okay. And is there any difference of the phenotype in the early onset or late onset COPD?
Early onset COPD have the late onset COPD have more emphysematous in radiology as compared to the early onset phenotype. Those patients who have late onset phenotype in radiology, if you see, there is more emphysema in the late onset phenotype as compared to the early onset.
Okay. Thank you. Is there any question from the audience?
There is a very good paper on Salvi, sir, probably. Between smoker and non-smoker COPD, one must go through this paper. This is a very nice paper. Sir has very beautifully described what is the difference between smoker and non-smoker COPD. Detailed regarding the radiology, clinical presentation, biochemical parameters. Everything is there. One must go through that paper. Very nice paper, sir.
Okay. If there is no question, then
Thank you, sir.
we can conclude the session. Thank you.
Thank you, Dr. Behra. Next, we have a workshop on iOS and FOT by Dr. Sandeep Salvi, Dr. Disha Ghorpare, and Dr. Shoibal Moitro. Sir, please come on the stage.
And I bring you greetings from our institute in Pune called the Pulmocare Research and Education Foundation, and we bring you greetings, myself and Dr. Sandeep Salvi, and we also welcome Dr. Saibal Moitra for the iOS and the FOT workshop. What you see on the screen is, this is how we've divided the session. We will have Dr. Sandeep Salvi first talk on the basics of lung function, which would be followed by a very interactive session on the basic principles of lung oscillometry, where all of us would discuss, have questions from you all on what lung oscillometry actually is, followed by a very short session on what are the prerequisites that as a doctor or as in your clinics, as your technician performs the lung oscillometry test, what are the things that are important that need to be taken care of. Then we break for lunch. As we are back from lunch, we would have a practical demonstration where we would have volunteers from your end, and we would actually perform the test and show you all and read the reports. Then we will have Dr. Sandeep Salvi talk on the principles of interpreting lung oscillometry reports, followed by a couple of case studies with Dr. Saibal Moitra, Dr. Parthasati Bhattacharya, and Dr. Sandeep Salvi. The last part would be a hands-on session where we would have devices put on, and you all can go on each of the desks and actually get your lung oscillometry test done and learn how to read the reports. So with this background, I would now like to invite Dr. Sandeep Salvi to take his session on the basics of lung function. Which one, this one?
Thank you, Dr. Disha, for those kind words of welcome and introduction. So wish you all a very good morning. I think it's good afternoon now. The whole objective of this workshop that we're conducting is to sensitize you, educate you, and empower you with the knowledge and the skill set for this very important diagnostic tool called oscillometry. There are several names that are associated with it: impulse oscillometry, force oscillation technique, lung oscillometry, airwave oscillometry, and so on. So all of them are essentially what I would call as lung oscillometry or respiratory oscillometry. So let's have the categorization of that name very clear. But before we get into that, I thought it's very important for all of us who are sitting over here to appreciate the beauty and the power of breath. Breathing is such an important activity, and I will set the foundation for the subsequent talks about the importance, the value of our respiratory health, how do we measure the lung health, and then, as I said, that will be the foundation for the oscillometry workshop. Life starts with the first breath, correct? A baby doesn't cry until it has taken the first breath. And life ends with the last breath. Isn't it fascinating that life is a period between the first breath and the last breath? Life is equated to breathing. If you're breathing, you're alive. If you're not breathing, you're not alive. That is how vital this simple function is for our survival and for our health. Out of the three basic necessities of life, food, water, and air, food and water are the ones that we give the most importance to. And the reason for that is we have to work hard for it. We have to pay money for it. If you want to eat good quality food, if you want to drink clean water, you need to spend money for that. And therefore you have value for it. On an average, one probably eats about 1 to 2 kilograms of food, breakfast, lunch, dinner, some snack in the middle. You put all that together on a weighing pan, it will be about 1 to 2 kilograms, right? The amount of water that we need to drink is about 2 to 3 liters during the 24 hours. But if you look at the third parameter, the third vital element, which is air, is the one that is perhaps the most widely neglected one. We take air for granted. Why do we take it for granted? Because we don't have to pay any money for it. On a daily basis, each one of us needs to breathe 10,000 liters of air to keep ourselves fit and well. And one of the most important ingredients of air that keeps us alive is this molecule called oxygen. So we breathe because through the breath, we nourish our body with oxygen. The oxygen that we breathe generates 90% of the body's energy. Did you know this? The food that you eat and the water that you drink contributes to only 10% of the energy that you produce every day. The air that you breathe or the oxygen that you breathe generates 90% of the body's energy. In fact, if you look at the composition of the human body, 65% of what you are is made up of oxygen. Did you know this? Obviously, a large part of that is because of water content. Water is H2O. And within the H2O, hydrogen is very, very light. Oxygen is the heavy molecule. So what you weigh, 65% of your weight is oxygen. Not only that, but the three basic elements of life, carbohydrates, proteins, and fats, all of them have oxygen within the structural component. So you cannot have a protein, you cannot have a carbohydrate, you cannot have a fat without an oxygen molecule. So oxygen is everywhere. Oxygen is there in proteins, carbohydrates, fats, water, everywhere. And therefore this vital element is the one that gives us life. And the organ that has been dedicated by nature or entrusted upon the responsibility of providing the body with oxygen is this organ called as the lung. The main function of the lung is to nourish the body with oxygen. We breathe 10,000 liters of air every day. 21% of that is oxygen. And out of that, the lungs, they soak in 1,000 liters of oxygen every day. So how much oxygen do we require on a daily basis? Around 1,000 liters. That's a lot of oxygen, isn't it? Compare that with the 1 to 2 kilograms of food and the 2 to 3 liters of water. We need 1,000 liters of oxygen. The other main function of the lung is to get rid of carbon dioxide. 350 liters of carbon dioxide is produced every day. And somebody has to excrete it. Imagine if you are excreting carbon dioxide through the urine, or you are excreting carbon dioxide through the feces. My God, you would be passing urine or feces all the time. So the lung has been given that responsibility of excreting 350 liters of carbon dioxide. And in order to perform this vital function, if you look at the architectural design of the lung, absolutely amazing. Two important components, the ventilation component of the architecture and the perfusion component of the architecture. Both of them work in tandem. 40,000 branching airways, 10,000 liters of air, and 10,000 liters of blood flows through the lungs every day. Isn't that amazing? One entire side of the heart is dedicated to supplying blood only to the lungs. That is how vital this function is. The oxygen is soaked up by the lungs through the alveoli, pushed into the blood circulation, and then it is transported to all the cells in the body by this carrier protein called hemoglobin. Hemoglobin carries oxygen from the lungs to the tissues. The hemoglobin is situated in the red blood cell. Every red blood cell has 300 million hemoglobin molecules. Amazing, isn't it? You know, you have to have 300 million hemoglobin molecules in order to transport the 1,000 liters of oxygen. And therefore, the red blood cell, in order to pack as much hemoglobin as possible, it has even thrown away the nucleus. Why? It wants to utilize that space to pack in more hemoglobin. So that is the only enucleated cell in the body, right? So oxygen is transported inside the red blood cell through the hemoglobin protein. Another amazing fact, absolutely amazing, and I did not know this for a long time. If you count every single cell in the body, from the tip of your hair to the tip of your toe, every cell, the heart, the lungs, the brain, the kidney, muscles, there are 37 trillion cells. Somebody has counted them. Can you believe it? 37 trillion with a T. Out of these 37 trillion, 25 trillion are the red blood cells. Isn't that amazing? 2 million red blood cells are produced every second in the bone marrow, and 2 million red blood cells are destroyed every second in the spleen. Look at the amount of investment that the human body has made in order to soak in the oxygen and transport it to all the organs of the body. This is the beauty and the power of our breath. 50% of the nutrient that circulates in the blood is oxygen. Did you know this? So if you take 100 ml of blood, put it in a test tube, around 45 to 50% will be the red blood cells. That is called as the hematocrit, isn't it? So 50% of the component of the blood is actually oxygen that is present in the red blood cells. So if you want to measure lung function, is your lung functioning well or not? To find an answer for that, my God, it's such a complicated answer because the lung does such amazing roles that there is no single measure of its function. Unlike your circulatory system, blood pressure, there's only one parameter, blood pressure. So what are the questions that we need to ask? Are the airways patent? Are the airways hyperresponsive? Are the airways inflamed? Are there adequate numbers of alveoli? Has the lung lost its elasticity? Is there fibrosis? Is the diffusion apparatus functioning well? Are the diaphragm and the chest wall functioning well? Is the pulmonary circulation functioning well? If you find the answers for all of those, then you will get to know the answer whether the lung function is good or not. So the lung is an extremely complicated organ, and therefore people have been trying to understand, find simple objective measures of the health of the lung. One of the first tests that was used to measure the health of your lung was the candle blow test. So you light 10 birthday candles, and if a healthy person could blow out all the 10 candles in one single blow, and the patient who is sitting in front of you can blow only 5 candles, what is that patient's lung function? 50%. There's a number you got, isn't it? It may not be the most informative number, but at least there's a number that you got to tell you how healthy your lungs are. So this was the first objective tool that was used to measure lung function. And then came the spirometer in 1846. Sir John Hutchison has been given the credit of inventing this very important lung function tool called spirometry. One of the parameters that he introduced was the concept of vital capacity. Take a deep breath and fill your lungs with as much air as possible, and then remove all that air out. The volume of air that you can remove now, completely out of your lungs, although there will always be some air in the lungs that you cannot measure anyways, but that volume of air was what he called as vital capacity. And you know why he called it as vital capacity? Because he was able to show through research that that volume of air was vital for survival. If you have bigger vital capacity, you will live for a longer period of time. If your vital capacity was less, you will live for a shorter period of time. Better predictor of survival than your blood pressure, blood sugar, ECG, anything, blood cholesterol, better than that. So your vital capacity decides how long you're going to live. And therefore he called it as vital capacity. Now, over the years, spirometry has then become a standard diagnostic tool. However, it took about 175 years for spirometry to establish itself. You know, 1846 was when the spirometer was first introduced. And in India, I can tell you, in India, we started using spirometry widely after the year 2000. And over the years, I think the use of spirometry has gone better and better. However, spirometry has a lot of challenges. One, the breathing maneuver that we perform while doing this so-called spirometry test is a forceful exhalation maneuver. And that forceful exhalation maneuver is not what we call as physiological. How do you measure a blood pressure? Do you ask the patient to keep on running and then while running you measure blood pressure? No. Do you do the same thing with your electrocardiogram or your blood sugar level? Eat a lot of sugar and then we'll measure your blood sugar. You have to do it at rest. So all the diagnostic tools are performed at rest, excepting this test called spirometry. It's a forceful maneuver. You have to exert yourself, and then only it give you reliable results. Requires patient cooperation. Not an easy test to perform. Even in the best possible hands, best possible technicians, only 35% of the patients will do good quality spirometry. Even in the most reliable hands, 65 out of 100 will not be able to do good quality spirometry. Cannot be done in everybody, like infants, uncooperative patients, elderly, those with severe disease, neuromuscular disease, and so on. The deep inhalation that you take before you do the forceful maneuver itself creates physiological changes in the lung. You know, in a normal healthy individual, if you take a deep breath, that deep breath stretches the airways and that itself causes bronchodilation. because there is a factor called as epithelium derived relaxation factor, which gets activated when you take a deep breath. Now that is giving you a physiological response. So it is not telling you the reality of what is happening. On the other hand, in asthmatics, if you take a deep breath, it causes more bronchospasm. So it amplifies the bronchospasm in an asthmatic patient, and therefore it is not a realistic test. Interpretation is a challenge for most physicians, and no good quality predicted equations in our country. And then more recently, we have started realizing that if you're a smoker, you may have smoked for 10, 15 years, you may have respiratory symptoms of cough and breathlessness, but in a significant number of them, you will find normal spirometry. But if you do a high resolution CT scan in these patients, you will find evidence of COPD. So spirometry doesn't pick up COPD in the early stage. It is not a sensitive tool to pick up early COPD. So these are all the challenges of performing spirometry. Now, over the years, we have understood the importance of lung volumes. And we have all been taught about this in the first MBBS, about lung physiology. The normal breathing that we do is called tidal breathing. Normal breathing. We breathe about 12 to 15 times a minute. That is called as tidal volume. That volume of air is called tidal volume. It's about half a liter, 500 milliliters. You can breathe up and above that, and you can remove up more below the actual tidal volume as well. So the up and above the tidal volume, that is called as the inspiratory reserve volume, and below the tidal volume, up to the base, is called as the expiratory reserve volume. That portion of the air that you cannot remove from the lungs all the time, always, is called as residual volume. So these are the capacities and the volumes. If you combine the tidal volume and the inspiratory reserve volume, that is called as inspiratory capacity. Functional residual capacity is below the tidal volume. The expiratory reserve volume and the residual volume together is called as the functional residual capacity. And I'll tell you, this is the most important physiological parameter that is important for us. Vital capacity is all that excluding residual volume, and total lung capacity is all that including residual volume. So these are all the important parameters that we need to be aware about. And as I said, from a physiology point of view, the most important spirometric parameter, or the lung function parameter, not spirometric, is functional residual capacity. Now, what is functional residual capacity? At the end of your tidal breathing, whatever air remains inside your lungs is called functional residual capacity. It's about 2.5 liters in females and about 3 liters in men. Now, it's a bag of air which is there inside your lungs all the time. 2.5 liters of air is always there. And on top of that, you take half a liter with each breath. So out of that half a liter, 150 ml is actually in the upper airways, which doesn't take part in the gas diffusion. So that's called as the, what is it called? Yes, and the remaining 350 ml is the one that actually mixes up with the functional residual capacity. Now, this is a very important slide that I'd like you to pay a little attention to. It tells you something very, very important. Look at the way nature has designed the lung and the chest wall. Beautiful. The lung is always tending to collapse inwards because of the elastic properties. If you give the lung a chance to do whatever it wants, it will always go inwards. And on the other hand, the chest wall always wants to go outwards. At FRC, at functional residual capacity, the inward pull of the lung and the outward pull of the chest wall are perfectly balancing each other. Okay, so functional residual capacity occurs when the two opposite forces are perfectly balanced. Now, what this means is that inhalation is the only time when you spend energy, because you want to open the elastic properties of the lung. So when you want to inhale, you have to spend energy in opening up the alveoli. Now, because the lung is such a wonderful elastic organ, it comes back to its original position just because of its physical structure, the elastic properties. You don't have to spend energy during exhalation. Did you know this? You spend only half of the energy in your breathing. That is your inhalation part. Exhalation is a passive process. You don't spend any energy for that. And the beauty of this whole structure is the lift. You have the lift and then you have a pulley that has weights perfectly balanced. So this improves the efficiency for the use of the electricity. If you want the lift to go upwards, you only need a kickstart. The other side is the weight that will take the lift up. Similarly, if you want to bring the lift down, you just need a kickstart. Electric charge only for the kickstart, and then the weight itself will bring it down. So the lift is a wonderful example of how the lung also works. When there are two opposite, perfectly well balanced things, you spend the least amount of energy. Okay, so I think it's very important to realize this. So efficient is that breathing process. We breathe 10,000 liters of air every day. How much energy does the human body spend on that? 1 to 2%. Can you believe it? I mean, look at the way the lung is designed. You breathe so many times in a day, 15 times a minute, and you multiply that by 60 into 24 times. You breathe so many times, but you spend only 1 to 2% of your energy because of the super efficiency of the respiratory system. And I think this is so amazing. The lung, as I said, is a very, very elastic organ. So imagine an elastic, if I have an elastic band, okay? The ability to extend that band, how much distance can it move? That is called as compliance. And the speed with which it comes back to its original place is called elastance. Compliance is the ability to be stretchable. Elastance is the ability to come back to the original shape as soon as possible. Now the lung is an extremely compliant organ and an extremely elastic organ. There is no tissue in the body that has got such properties. It has got compliance also, it has got elastance also. So this is an example of, there's a spring, I put some weight over here, perfect elasticity, compliance and elasticity. And therefore, if you see functional residual capacity is perfectly balanced by the elastic recoil of the lung and the chest wall elastic recoil. Both of them are in the opposite direction. FRC is right in the middle. Now, when the compliance increases, for example, in COPD, the spring becomes very loose, easily goes down, but doesn't come back to the original position. That is called increased compliance, reduced elastance. Okay? So when that happens, for example, in emphysema, then the chest wall becomes more powerful, and then the chest takes power over the lung. That's why you get hyperinflation. And the third one is the spring becomes very, very tight, very difficult to move it. And the moment it moves, immediately it comes back to normal. Poor compliance, increased elastance. And the typical example for that is pulmonary fibrosis. And in pulmonary fibrosis, the FRC then goes towards the lung side. Okay, so I think this is important from an lung oscillometry point of view. Just to highlight, the connection between the lung and the tree. Both are the same images. I've just inverted one. One is the lung and one is a tree. The inverted image is the tree. The lungs and the trees, they look exactly similar, very similar. They have a central branching, leaves are the alveoli, and so on. The lungs take in oxygen and give out carbon dioxide. And the trees, what do? They take in carbon dioxide and give out oxygen. What better example than made for each other? The lungs and the trees are made for each other. What a beautiful example, isn't it? The other beauty and the efficiency of the tree and the lung is given over here. Have you seen tall trees? They can grow as tall as 100 meters. And my God, it's about 30 stories, 30 to 40 stories. Some trees can grow very tall. Now imagine the amount of energy that the tree must be spending in transporting water from the soil to the tip of the tree. 30 stories. You probably need a very powerful pump, isn't it? You need a very strong motor. How much energy do trees or plants spend for moving water from the roots to the top? Any idea? Where do they get this energy from? I think the answer lies in this simple law of physics. Water evaporates at what temperature? Yeah, water evaporates at what temperature? 100 degrees? Yes, water evaporates at all temperatures. Did you know this? You keep a cloth, a wet cloth for drying, even at 10 degrees Celsius. It will take a long time, but it will evaporate. Remember that water evaporates at all temperatures. Simple law of physics. And the trees have used this simple law of physics. The energy that they spend in transporting water from the roots to the top is zero. Trees don't spend any energy. They use the power of evaporation. On the leaf, there's a stomata through which evaporation happens. That evaporation pulls the water column upwards. The tree does not spend any energy in moving the water from the soil to the top. Look at the efficiency of the tree. And the same efficiency has been given to the lungs as well. Now, when you have to move a gas column from the mouth to the alveoli, you have to overcome certain hurdles, certain resistances over here. Now, where are those resistances? The nose. The air has to go through the nose. 10,000 liters of air, remember that. 500 ml through each breath. 20% of the resistance is offered by the nose. The chest wall also has to expand. 46% of the energy is consumed over there. And within the respiratory tract, the large and the medium sized airways, they contribute to 20% of the resistance. Small airways contribute to 7%, and the lung parenchyma contributes to 7% for the elastic properties. What is the main message I want to tell you over here? The small airways, they contribute to only 7% of the total respiratory resistance, only 7%. Why is it important? Because if you have a disease that primarily affects the small airways, it will not create any symptoms for you because it is not contributing to the resistance. The other way of understanding that is, until a significant amount of the small airways get damaged, you will not start manifesting with symptoms. By the time you manifest with symptoms, you have already 50% of the small airways are damaged. It's too late to do that. Have you all understood this? Small airway dysfunction is a very, very important component of our disease profile pattern. And if you look at the branching pattern of the airways, on the x-axis over here are the generations from trachea is 0, right main, left main bronchus is 1, and then subsequently it divides 23 times until it reaches the alveoli. Right? So in these 23 generations, the highest resistance in the respiratory tract is at the 6th generation. From the 7th generation onwards, the small airways start. Small airways have an internal luminal diameter of less than 2 millimeters. The 7th generation starts, the small airways start at 7th generation, and then the resistance falls down as we go to the 23rd generation. Okay? Why am I talking about small airway dysfunction? If I ask you this question, what portion of the airways are affected in asthma? A is the large airways, B is the medium sized airways, and C is the small sized airways. So in asthma, which airways are affected? Is it A or B or C or a combination of all the three or predominance of one? Which one? Sorry? Small airways. Small and medium. Large and medium or small and medium? Small and medium. Spirometry is a tool that does not pick up small airways easily, and therefore we have never known what is the contribution of small airways in asthma. To cut the long story short, there's this big review article that was published about 5-6 years ago. What proportion of asthmatics have small airway dysfunction? Answer is 50 to 60%. What proportion of COPD have small airways dysfunction? 100%. COPD is a disease of the small airways. 50 to 60% of the asthmatic affect small airways, and these are not easily picked up on spirometry. Why is this information important? Because if you have small airway dysfunction asthma, it gives rise to a very unique phenotype of asthma that is very difficult to treat. Poor control asthma, difficult to treat asthma that requires SABA use all the time, asthma that requires oral corticosteroid use. So if you have asthma that affects small airways, it's the difficult to treat asthma. So there's this review article that was published 2 years ago called SAD asthma. What is SAD? Small airway dysfunction asthma. Asthma that is predominantly affecting the small airways. Difficult to treat asthma, more severe form of the asthma, more uncontrolled asthma, nighttime awakenings, obesity-induced asthma, exercise-induced asthma, smoking-associated asthma is all small airways. Why is this important? From a pharmacotherapy point of view, if you have a disease that is predominantly affecting the small airways, you have to give drugs that reach the small airways. The current drugs, salmeterol, fluticasone, budesonide, formoterol, do they reach the small airways? They don't. And maybe that is the reason many asthmatics are not getting better on the drugs, because they're not reaching the small airways. Does my asthma patient have small airway dysfunction? Spirometry doesn't give me the answer. The FEF 25 to 75 gives you some clue, but it is not as good as what other tools can do. So that has become a big thing. The future of asthma, I can tell you, is going to be this phenotype called SAD asthma. If you have an asthmatic in a clinic who is not doing well, you have given the best possible treatment, you have avoided all the triggers, patient is taking the inhaler correctly, patient is also adhering to the treatment correctly, still the asthma is not getting controlled. It is very likely that they have SAD asthma. Now, how do you diagnose that? Spirometry doesn't do that. Now, SAD asthma has been shown to be very, very common. And if you look at small airway dysfunction and asthma, if you have predominant small airway dysfunction, then you fall into the category of poorly controlled asthma. So SAD asthma should be added to the list of risk factors that are associated with poor asthma outcomes. COPD is a disease of the small airways, no question about it. So spirometry doesn't pick up COPD in the early stage. HRCT shows COPD, but the spirometry is not showing COPD. Why? Because spirometry is not very sensitive to pick up small airway dysfunction. Now it is with this background that I want to introduce to you all this whole new tool called oscillometry. If you look at the algorithm for spirometry, and I'm sure all of you all are well versed with it, if your FE1 upon FVC ratio is more than 70% and your FVC is more than 80%, we call this as normal spirometry. If your ratio is less than 70%, then this is obstructive airway disease. If the FVC is more than 80%, it's mild or moderate. And if the FVC is less than 80%, it's severe obstructive airway disease or a combination. Now, for all these years, we were categorizing those patients who have a ratio of more than 70%, but FVC less than 80% as suggestive of restrictive lung disease. That was a term that we always use in our reporting. Now we have started realizing, it may not necessarily be restrictive lung disease. Because if you do body box plethysmography in all these patients, only 40% of them show true restrictive lung disease. What about the remaining 60%? They are the ones that we call as PRISM. What is PRISM? Preserved ratio impaired spirometry. The ratio is normal, but the FVC is less than 80%. And this is the beginning of small airways obstruction. This is the beginning of asthma. This is the beginning of COPD, which was never categorized in the obstructive airway disease phenotype. Have you all understood this? So the pattern that looks like restrictive lung disease is actually early obstructive airway disease. Now, I'm not going to go into the details of this, but if you follow these PRISM patients over a period of time, half of them will develop COPD. So it is a pre-COPD. It tells you that these patients will develop COPD over a period of time. The lung oscillometry is perhaps going to be the future of lung function testing. Why perhaps? I'm confident that it will be the future of lung function testing. It has several advantages over spirometry. First and foremost, it's a very simple test to perform. Unlike spirometry, where you have to do a forceful exhalation maneuver, here you do a normal tidal breathing, which is physiological. You understand what is happening at the FRC component in your lung function. Spirometry doesn't do that. The test is very simple to perform. It takes only about half a minute to 1 minute to perform, and more importantly, it is very sensitive in picking up small airways dysfunction. So whether it be early asthma, whether it be early COPD, this is going to pick up early diagnosis better than spirometry. Now, if you look at the applications of oscillometry, there's a whole host of them. I'm not going to read all of them, but we have started appreciating the value of this tool. Lung oscillometry is not only for obstructive airway disease, it is also useful in restrictive lung disease, and it is also useful in a whole host of other conditions. So, more value for money than spirometry. Oscillometry was invented in 1956. Why suddenly are we waking up to it? Why after so many years? The same thing happened with spirometry as well. Spirometry started in 1846, but it took 175 years for it to get established. So historically, if you see, people don't accept diagnostic tools very easily. Now here is a very nice commentary written by Professor Peter Calverley, one of the most respected names in the field of COPD research, from Liverpool University in the UK. Now he wrote this wonderful article, Oscillometry, Old Physiology with a Bright Future. And he tried to answer this question, why has lung oscillometry remained a neglected entity? And then he gives all the reasons. Intellectual inaccessibility. It took such a long time for us to explain to people what is vital capacity, what is FEV1, what is FEF 25 to 75. It took us a lot of effort and hard work to explain those concepts. People started understanding that. But then suddenly you're telling, no, oscillometry doesn't tell you FEV1 and FVC values. It tells you resistance, reactance, impedance. Now, what is reactance? What is impedance? For doctors, it's a challenge. Off-putting for the less numerate physicians. Lack of standardization. So the predicted values not very well established. And then interpretation has been a challenge. The only company that was making oscillometry was Jaeger, and now it's called Wire. So they were marketing the impulse oscillometry for many, many years. Unfortunately, they did not contribute to knowledge dissemination to the doctors. They were selling the machines to all the big organizations, big hospitals, but nobody knew how to use it. Nobody knew how to perform the test. Nobody knew how to interpret the test. Now, there are so many people who have come into the market. So, there are 10 oscillometries that are available in the market just in the last 3 to 4 years. Why? Because of the realization that this is going to be a very important diagnostic tool. So, exciting times for all those of you who are sitting here. If you're looking for new things to start in your clinic, one of the first things that you should most certainly think about is think of having an lung oscillometry system in your clinic, because it is going to be the future of lung function testing. And this workshop is going to educate you about what this is all about. There's a very beautiful review article written in the European Respiratory Review just a few months ago. Please read this article. It will give you everything that you need to know about oscillometry. And then before that, there are standards for respiratory oscillometry, which was published in 2020 in the European Respiratory Journal. Standard methods for doing oscillometry. So as I said, exciting times, likely to be the future of lung function diagnosis. And therefore, you're very lucky to be sitting here and listening to something that will be the future. So with this background, may I now ask Disha to come ahead and take the next, what is the next session? And if there are any questions, I would be more than happy to answer. Any questions, any comments? Yes, ma'am.
As you mentioned that when there is a problem with the small airways, we do not really have symptoms related to that. So is there any particular symptom that you know of which might be an indication of small airway damage?
Shortness of breath. This is the first
that is the
symptom.
only.
Shortness of breath. But shortness of breath can be because of so many other conditions. You'll never know that. So shortness of breath on exertion becomes the first symptom. But then if you do in your routine day-to-day life, unless you start running, And then only you start feeling that shortness of breath on exertion. So people don't capture it. And shortness of breath, anemia, heart disease, so many other conditions. Therefore, it's sensitive but not specific. Yeah, any more questions? Any more comments? Any comments? I hope you all have understood the beauty and the power of breath. It's so important, as pulmonologists, as chest physicians, it is important for us to understand and appreciate the organ, the beauty of this organ. It's not there in the textbooks. All the information that I've given you they're just pearls of wisdom that I have collected over the years and I've shared them with you.
Thank you so much, sir. Now I would like to invite Dr. Saibal Moitra to take the session ahead on the basic principles of lung oscillometry.
Yeah, thank you. First of all, I would like to thank the organizers for organizing this extremely important workshop and inviting me into this team. I can understand that you all are spellbound by what Dr. Salvi just spoke. Okay. But yes, it is very true that oscillometry is going to be the future tool in the lung function or the airway disease assessment. And we all need to understand it extremely well, that why do we need it, what are the advantages, what we measure, how do we interpret the results, and this translate it into a clinical decision making. So since you have understood that what is spirometry, means what are the various actually the challenges which there is spirometry which are not met, or the unmet challenges in the spirometry, and what are the other issues that cannot be answered by a spirometry. The most two, three things that Dr. Salvi has said is, first of all, it's a non-physiological method of assessing a lung function, and it cannot pick up the small airway dysfunction early. So means these are the main two important things. And third important is that you need a lot of patient cooperation in doing the test. So that means if your patient cannot cooperate well, so that means you cannot do that test. So it could be a child, it could be an aged person who has some cognitive impairments, or there are other impediments in doing a good forced expiratory vital capacity maneuver to get an objective assessment of the airway diseases. So now I will just go very slow. As Dr. Salvia said, that it takes time to understand these parameters which are being measured by the oscillometry. So we need to know that what these parameters actually are and what they are telling us and what they are actually capture in while you are doing this test on the patient. So once you have this knowledge, then you go into what are the algorithms for the interpretation, how do you do the test, and then you yourself perform the test, interpret the results. Okay. So just repetition, that this is a technique which basically uses sound waves. Now, there are other applications of sound waves. You know that it has been there in the clinical medicine. This is another application of sound waves, where these sound waves are actually thrown into the respiratory system, and again they come back, they're reflected back, and then these sound waves are actually some parameters in the sound waves, they are measured to give rise to the various metrics. So basically it is one of the application of the sound waves in the respiratory medicine to know about the details of the lungs, as the airways and the tissues and all. And as you have seen, that it's an old physiology with a bright future, So it was given very long back by Dubois in 1956. And you know, this Dubois gave us another very important lung function instrument, which we use very commonly. Can anyone say what is that? Yes, body box. But the utility of the body box, we have been using it more. We came to understand better way. Why? Because we can actually understand it in a much easier way because of the training in the lung physiology which we get during our first year MBBS days. But we are not trained in the way to understand the oscillometry. We did not have the training in our like medical formative years. Yeah. So that's why it comes as a thing which is pretty new, maybe Greek. So, and we just fail to understand it. So for myself, when I started working with the oscillometry, really for me, also it took a lot of time, like several months up to 1 year, to actually understand that what is actually it is and what is it's saying. Okay. So now that, yes, so now the sound waves which are being sent into the respiratory system, and depending upon the sound waves, the type of the sound waves being sent, we have various types of machines. So the interpretation and the result that they all give is similar. It's only the type of the sound wave that is being thrown inside that decides what that is actually different in the different machines. Now, to be very to say in nutshell, the sound waves it could be a monofrequency sound waves. You know, sound wave is a sine wave. Okay. So it can be of a single frequency, say of 5 hertz sound wave, it goes inside, comes back and interpreted. So at a single time, only a single frequency sound waves goes inside, comes back, you interpret the results. Again, another frequency sound waves goes inside, comes back, you interpret the results. That means at a single time, it's only a mono frequency, a single frequency sound wave is going, where the machine is throwing a single frequency sound wave. So that can be the one type of machine. There can be another type of machine which is actually throwing many sound waves at the same time. But you know what is a noise? Means you must have been to a marketplace. Okay, so we all are familiar with noise. There's so much noise around us. Okay. Noise is basically when at the same time there are multiple frequencies of sound waves are coming and they're being produced by many sources, multi-frequency sound waves at the same time, and they do not have any relationship with them. That is it's just happening like that. So all randomly distributed sound waves from the various sources, they're coming at a particular same time, and it makes a noise. Okay. You must have heard some concert, like any musical concert you must have heard. So there also they produce the sound waves from the several instruments, okay, at the same time. But is that a noise? No, they have a cadence to it, and there is a tempo, and there is a temporal credibility to the entire sound waves, which gives the music. So we do not say it's a noise. So there is a particular tone, there's a particular amplitude, there is a particular way in which all the musics play, or all the instruments play. But in noise, there is no such correlation between any of the sound waves produced by any of the various sources. So it's a noise. Now, what is this pseudo-random noise? Pseudo-random noise is basically, it looks like to be a sound waves which is quite random. That is, say 5 hertz, 10 hertz, 15 hertz, 20 hertz, all these sound waves are being produced at the same time, and they get mixed up, and they again go into the respiratory system and again come back. But actually, it is not random. Actually, there is also a minute difference between first 5 hertz goes, 10 hertz goes, 15 hertz goes, and comes back. So there is a temporality of the sound waves which is being thrown into the system. So though it looks like to be a random noise, but it is not. So it is called a pseudo-random noise, oscillations or PRN oscillations. So there are various instruments which is based on the sound waves are actually pseudo-random noise sound waves. So one of the instrument is this air wave oscillometry or the tremoflow. This is the instrument there. Now came another very important improvement on that. So we as a clinician, we do not need to understand all the nitty-gritties of how the sound waves are produced and what is actually meant by the impulse oscillations. We can have only a vague idea about it. What is this? Now it was this impulse oscillations or the impulse oscillometry. This was a development on the acoustics or the type of the sound waves in the way it is being thrown into the system, and which came pretty later. Like it was given around in 1971 by Michelson. Now what they did is that, so there is a concept which was a mathematical concept which is called a fast Fourier transformation. We do not need to know what is a fast Fourier transformation. It's a computational concept. What it did is that it just collated all the frequency sound waves into a packets. Okay, so that means there is a single packet which consisted of sound waves of 5 Hz, 5 to 35 Hz. All the frequencies were integrated by this fast Fourier transformation method, and they form a square wave packets. And these packets of the sound waves were then thrown into the respiratory system at a particular frequency of 5 Hz. Okay, so this is how it was done. Now why it was done? Because actually it helped in some of the ways in improving what they call the signal to noise ratio, and they helped in understanding in more detail in what are the other dynamics, okay, and make the machine many more sensitive. But it has its own disadvantages also. All the machines has but we need to understand that these are the packets which are thrown. So whether you are using an impulses of the sound waves or the pseudo-random noise sound waves, the monofrequency sound waves we hardly use these days because it takes time. So whatever we use, it actually gives the same result. So it does not matter. So whatever it's using, it's only how you understand the machine well, how you understand the concept well, and how you can use it more efficiently on your patient is what matters. Okay, this is the data. If you want me to repeat anything, or if Dr. Salvi wants to add on to it, or Disha wants to add on to it, we can just have it as a discussion forum. Okay, so it may not be too monotonous like a didactic lecture. So this is what I was talking about, the fast Fourier transformation, and these are the square waves of the sounds, which is packets of the sound wave which is being thrown inside. Now how this synthesis is done, the Fourier synthesis. So this is actually means that you can see there's the various frequencies of the harmonics is there. That is the basic harmonics, and the number of frequencies increase. All these are then integrated together to produce the square wave harmonics. That is the sum wave. Now this is what is being thrown inside. Now this is that on the right hand side. That is actually a sound wave. Okay, so sound wave it's a pressure wave. It's a pressure wave, and its frequencies is varying. So that means you can have a pressure wave, sound wave of 5 Hz frequencies, which is actually oscillating at 5 times a second, or it could be a sound waves of 20 Hz. Okay, the frequency could be 20 Hz. So more higher the frequencies, the sound waves are actually beating that fast. Okay, so that is it. Now what is this one? And this is actually the time amplitude of the normal tidal breathing. The normal tidal breathing is 16 to 20 breaths per minute. Now, so when an individual is breathing 16 to 20 breaths per minute, if you can just transform it into the time amplitude curve, you get things like this. So this is the normal tidal breathing, and those are the sound waves of maybe of 5 Hz or 10 Hz or 15 Hz or 20 Hz, whatever it is. Now what is being done in the system is that these sound waves are actually superimposed on the tidal breathing. That means the person is actually breathing normally, and then you throw the sound waves. So the normal breathing is actually a type of, generates a pressure and generates a flow in the air column inside the airways. Now these sound waves, they are also pressure waves. So that means any pressure wave will actually produce a flow in of the air inside the air column inside. So that means there is this sound wave which is also generating some pressure flow changes in the air and there's a tidal breathing also, which is actually also causing the pressure flow changes, which we understand very well. That's why how we inhale and exhale. Now these sound waves are actually superimposed on the tidal breathing, and this composite thing goes inside and it comes back. Okay. So what this machine does, this machine has a very beautiful signal filtering mechanisms. Okay. By the signal filtering mechanism, it can differentiate the pressure flow changes of the air produced by the tidal breathing on one hand, and on the other hand, the pressure flow changes which is being imparted because of the sound wave. And this, by the signal filtering mechanism, so pressure flow changes which is imparted by the sound wave, the machine can capture it separately and separate it from the normal tidal breathing. And then it gives a relationship, and from there we derive all the variables that we need to measure. That means the machine is doing a beautiful thing. It is sending the sound waves, the varying frequency, maybe by a pseudo-random noise oscillation or an impulse oscillation. It is coming back. The pressure is measured by you have the volume and the flow. This comes back. So it has been, flow is measured by the pneumotachograph, and we have the other transducers to measure the volumes or the flow or the pressure. And then it separates, the machine separates the signals which is coming due to the sound waves and the signals which is coming because of the normal breathing, and it gives us the result. So this is how this machine is doing everything for us. So, and this is how it is being interpreted. So sound waves going in, coming back, signals getting separated, and we get the pressure flow relationship depending upon due to the sound waves, and we find out the various parameters because of that, right? So sound waves are mini pressure waves. They will cause subtle pressure changes. Subtle means extremely subtle, minuscule subtle. The subtleness is to the tune of it's minuscule. Okay, and these changes are actually then measured by the machine. Now we should understand 2-3 important concepts. Now what are they? See, you have a sound wave of a particular frequency, low frequency, say 5 Hertz, and you have a sound wave of higher frequency, say 20 Hertz. So a sound wave, the pressure wave, so any pressure wave will have an energy in it. Okay. So if the sound wave is actually, it is vibrating slowly, that means it's a 5 Hertz frequency. That means whenever any sound waves vibrate rapidly, there is a particle collision between the various particles through the medium in which it is flowing. And this collision of the particles, it actually dissipates the energy which is stored in the sound wave. So if a sound wave is vibrating very fast, like if it is a high frequency sound waves, that means the energy dissipation is very large. So what happens is that a very highly moving sound waves is actually, the energy dissipation is very, very large, very fast. At least if you are running running, so those who is not a runner, so that means you're running, after some time you'll get tired, you will stop. But you are walking, maybe you'll be able to walk a larger distance, then you will stop. Similarly over here, so like the sound waves of the larger frequencies, they dissipate the energy fast. So when they've been thrown from the loudspeaker in the mouth, they will traverse some distance inside the lungs. And after that, the energy will just drain out of it and it will get dampened. So that means that sound wave cannot go very far. But if you have a sound wave which is of a smaller frequency, say 5 hertz, that means you're walking. It's actually walking. It's a walking sound wave. 20 hertz is a running sound wave. You can just think it like that. Okay. That walking sound wave can walk a long distance. Okay, it will go quite far, then it can stop. Okay. So that means those sound waves with a low frequency, the energy dissipation is quite less. So that means those sound waves do not get dampened, okay, quite easily. So they can traverse a long distance and then they might get dampened. For all clinical utility, we use sound waves usually between 5 hertz to 20 hertz in the majority. So if you say take a sound wave through a sound wave of 5 hertz, that 5 hertz sound wave, if you throw it at the mouth, that is going to go deep inside the lungs, and then it gets reflected. Some gets reflected and some gets transmitted and all these things. The reflected wave comes in it and is being sensed by the transducers as at the mouth. So that means that sound wave which has reached the periphery of the lung, that is a 5 hertz sound wave coming back from here, will give us the information of the entire system from the mouth till the periphery. And if the sound wave of 20 hertz, okay, which gets dampened, say, in the larger airways, so the information which the reflected part of that sound waves gives us will give us information only of the larger airways. This is a very important concept to understand. Lower the frequencies, they traverse farther. Higher the frequencies, they get dampened in the larger airways. So now this is just a schematic diagram of the oscillometer. I think, yeah. So means here you can see that means that a 5 hertz sound waves goes deep down, comes back and gives us the measurements of the entire system. The 20 hertz sound waves gets dampened in larger airways, so gives information about only the larger airways. So that means if you can mathematically deduce what is between the 5 hertz to 20 hertz, so that difference is actually, it is giving the various knowledge about the parameters of the smaller airways only or the peripheral airways. Okay, so 5 hertz entire system, 20 hertz only the upper airways, and maybe 5 minus 20 is only the peripheral airways. So this is how it goes. Now let us come to understand that what parameter we measure. So this is, yeah. So first parameter which we measure, complete parameter, is what is called an impedance. Now what is an impedance? Impedance is basically, first of all, understand that it is a sound wave, which is a pressure wave, which is being thrown inside the airways. That pressure wave is going to cause the movement of the air molecules, which is there in the air columns in the airways. So it is going to generate a flow. Okay, so whenever there is a flow of a fluid through any tube, that means that flow can get impeded by various factors. So any factor which is causing this impediment to the flow, the air, is basically the composite thing is known as the impedance, and we say it as Z. So that is the main composite metric we get by the virtue of using a sound wave is the airway impedance. That means, or we call it also respiratory system impedance. That means the impedance is basically, it is an impediment of the flow of the molecules, and this flow is generated by a pressure wave, that is the sound wave. So that is one. And this impediment, you can also say that impediment is somewhat synonymous, somewhat in question mark, to resistance. So it's not, everything is not resistance. So resistance is easier for us to understand because we all know Ohm's law, Resistance is pressure divided by flow. So that means, and you have seen in the previous slide that was presented by Dr. Salvi, is that the resistance which is offered to the flow of this molecules or a fluid or a gas, so is actually directly proportional to the length of the tube and inversely proportional to the fourth power of the radius of the tube. Okay, so that means the larger the caliber of the airway, less is going to be the resistance. So this is one very thing which is very easy to understand. But again, remember one thing: that whatever thing we calculate in an oscillometer has to be a function of the frequency of the sound wave we use, because the 5 Hz frequency is going to give some information of the entire system. 20 Hz frequency is going to give us information of the larger airways. So if we say the impedance, but respiratory system, we say the resistance, but that also has to be what is the frequency of the sound wave which was used to get an idea of this resistance. It's very, very important. So then we get to understand that what information it has given to us. Okay, so this is the total airway is by the 5 Hz. 20 Hz is the proximal airways, and peripheral airways is the R5 minus R20. So this is a very important concept we first need to understand. So this is the same thing, that impedance is a measure of opposition to the motion of a structure subjected to a force, and this is actually the entire respiratory system impedance which is being given, and also it is being written as omega. Now see two important things. Now let us come talk in little detail about it. This entire respiratory, because these are the parameters you measure in the oscillometry, and so we need to know what are the basics behind it. Now the impedance, you see, it is actually a summation of two another metric which is actually the subcomponent of the impedance. One is the R, or the resistance, which we just talked, and another is an X, which is a reactance. This was actually the most difficult part for I felt myself to understand. Okay. Now, what is this reactance? That we'll see. Always understand that any impedance is a function of the frequencies omega, which is given by the 2 pi f. We did not know to know about it. Only understand whatever we measure, it has to be a function of the frequencies. Now, resistance we have talked. What is the resistance? So resistance is what is given and by the pressure divided by the flow, and it is a function of, definitely, because of the frequency we have to measure. Now, one very important other thing we should understand while we're measuring the resistance, that is, in an adults, as Dr. Salvi said, we have seen that the smaller airways actually they contribute very less of the total airway resistance. It is mainly the upper airways. So basically, when we are throwing the sound waves, this goes inside and it comes back. So in a normal healthy adult, it's the small airways, they hardly produce any resistance. Okay, so it's mainly the larger airways. So whether you are giving the sound wave of 5 Hz, which is going deep inside and coming back, or a 20 Hz, which is going only proximal airway and coming back, the resistance does not change much with the change in the frequencies of the sound waves used. So that means in adults, the resistance in normal healthy adult, resistance is not a function of the frequencies. It remains more or less same throughout from 5 Hz to 20 Hz. But in children, it's something else. In children, and a large part of the resistance is being contributed to the smaller airways. So in children, if you throw a sound wave of 5 Hz, it is going to give a more higher resistance than the sound wave of the 20 Hz, even if the child is healthy. Okay. So that means in children, a frequency dependence of the resistance can be normal, physiological, but in adults, usually the normal physiologically, the frequency, the resistance is not dependent upon the frequency of sound waves, whatever you are throwing inside. So now, so that means
If you can see that in healthy adults, the resistance at 5 Hz and 20 Hz, they are nearly equal. If there is a central airway obstruction, that is going to be picked up by both the R5 because it's traversing the entire airway, and also by the sound of 20 Hz. So that means in central airway obstruction, both the resistance at 5 Hz and 20 Hz increases. So this independent of the frequency. But what happens if there is only a peripheral airway obstruction? That means all the sound waves, the 20 Hz or higher sound waves, which can only traverse the proximal airways, they won't be able to pick up the changes in the peripheral airways. So that means resistance at 20 Hz is going to be more or less normal. But the 5 Hz sound wave, which goes deep inside and comes back, the resistance increases very much. So if you just have to plot at a curve where actually the x-axis there is the frequency of the sound waves, and on the y-axis there is a resistance. That means at the 5 Hz the resistance is going to be higher, at 10 Hz, at 20 Hz resistance is going to be normal. So there's a curvilinear curve. Okay, that means it's going to be, it's called a frequency dependence of resistance. So in an adult, a frequency dependence of resistance is indicative of peripheral airway obstruction. So if there's a total airway obstruction, that means all the peripheral and the proximal airway obstructions, everything will be enlarged. Well, the resistance at 5 Hz would be enlarged much more than the resistance at the 20 Hz. So this is the curve which I was talking about. So the predicted line, so that is basically the normal we see in normal healthy adult. The resistance does not change much if it's a 5 Hz sound or a 20 Hz sound. But in case of a diseased individual where there is a peripheral airway obstruction or disease, the 5 Hz resistance goes up and we get this curvilinear slope of this curve. So this is the same thing, we're going to see more about it when we talk. Now let's understand what is reactance. Now see, reactance, now just pay attention to this. This is a difficult part. Reactance is what is called a rebound resistance. Now again, the reactance is made up of two further, there are two components to it. One is the inertance and another is the capacitance. You just have to rethink of what Dr. Salvia said regarding the stretchability and elasticity. Okay, we'll come to it. Say this is actually a say there's a ball on the table. Okay. Now, if you just try to push the ball, okay, then the ball will move, start moving. Now, if the ball is a light ball, means it does not have much weight, okay, you give a small push, the ball starts rolling. But if it's a heavy ball, okay, then what will happen? You have to give more push to make the ball rolling. So that means the amount of the pressure which you need to make a molecule move is actually dependent upon the weight or the mass or the inertia of that substance. So those substance which has a high inertia, you need to give a much greater push to make it move. Those who have a low inertia, you need less amount of force to make it move. Okay. Now say if this sound wave is moving through the air column in the airways, now there are molecules of the air, oxygen, nitrogen, carbon dioxide, all this, nitrogen, oxygen. So if depending upon the inertia of those molecules or other substances in that air which we are breathing in and out, so it also causes an impediment to the flow of that sound waves or flow generated by the pressure wave. So that means it is also actually resisting the flow of the wave, of the sound wave, because of the inertia of the molecules inside. So this is what is called an inertance. That means impediment to the flow because of the inertia of the molecules inside the tube. This is one. Along with that, there is another component, that is the capacitance. Now just remember what Dr. Salvi has said before, that is, lung is an elastic organ and it is also highly stretchable. So that means this is the tube, the air is, the sound wave is moving through it, it's a pressure wave generating a flow. Now when in a tube, the sound wave is moving through it, it is going to impart pressure all around equally. Okay. And that pressure is going to stretch the wall, that is the wall of the tubes, that is the airways, and cause it to deflect from its original position. So that means it's going to stretch it. And that stretch is what is called a compliance. Okay, so or the capacitance, that is, it's going to stretch it. So more or like compliant the tissue is, more it will be stretched. And once it has got stretched, it has an elastic property also that we have seen. It's going to come back. So that means it got stretched and it came back because of the elasticity. And when it is coming back, it is going to cause a little bit of pressure on that flow wave. So that means, so there is the flow, the sound wave which has generated the flow. It has caused a stretching of the airways and it came back because of the elasticity. When it came back, it put pressure on the flow. So that means, and this pressure, that is also is going to cause an impediment to the flow. But interestingly, it is because of the property of the tissues around it, which is called elastance. So that means this is another component which is also causing an impediment to the flow of the sound wave generated flow. Okay, but actually these are very minuscule. These are the impediments. So that means one is the inertance or the caused by inertia of the molecule. Another is the capacitance or the compliance or the elastance, which is because of the property of the tissues around it. And because of these properties, both of these is also causing an impediment to the flow of the sound wave. So these two add together and they give what is called a reactance. And these then add with the resistance and they give what is the impedance. So this is what is actually the reactance, which is a two component, inertance and the capacitance, and another is the resistance. So that's why reactance has also been termed as an imaginary resistance, but it's not an imaginary thing. It's so minute, so minuscule, that it can only be picked up by the oscillometry. No other device can pick it up. So that means they said it imaginary, but it's actually not an imaginary thing. It's what is there? It's there. It's true. So now, what is this capacitance? See, there is a convention to it. Now, this capacitance, I have said that this is because of the elastic property of the tissues, which is coming back and pressing on the airways. And since it is moving in a direction opposite to the pressure wave which was generated by the flow of the sound wave, it is opposite to it. No, it's coming back. Okay, so it was by convention, it was taken as negative from the baseline. It is written as negative. By convention, it's negative. So it is actually inertance I minus C, which actually gives rise to the reactance. Okay, so this is the concept of the inertance and the capacitance. So this is what is stated over here, that is inertance is because of the object's capability, because of its inertia to not to move from its place, and thus it gives inertance. So this is the relationship between the inertia and mass. So this you all know from your school days. I do not need to reiterate it. I just try to put it forth in what we measure. Yeah, so this is one of the important things. Now see, what happens in the large and the medium airways? In the large and the medium airways, the column of air is much large. Okay. In the smaller airways, the column of air is very thin, very less. So that means that the large and the medium airways, they have much higher inertance than the smaller airways because of the column of air is thin. Now just tell me that of what you understand from this, how useful is inertance in our clinical practice? How useful it is? Is it very useful or is it of not much use? It is. Why do you say it is useful? You are using this instrument to measure what? Are you using this instrument to measure the air what is inside, or are you using this to measure what is the nature of the tissues, the airways, okay, or the parenchyma, whatever you say. So that means if you're this an inertance is a property of the gas material inside. You are not much interested to know about it. In the smaller airways at least, you're not interested to it, okay. Though it is an important parameter which adds on to the capacitance to give the reactance, but for clinical purposes, we do not need the use of the inertance. So that means if we just want to see about the peripheral airways, where basically we are using very low frequency sounds, there the inertance is nearly zero, because the column of air is very thin over there. So whatever information we get about the peripheral airways by using a sound wave of low frequency, basically or majorly gives the information about the tissues, that is the airways, and also the tissues around it. So interesting, no? So that is the beauty of this instrument. Okay, so capacitance we just talked about. So elasticity and this is capacitance, they are just opposite to each other. You have seen that pulley over there. So its compliance is actually inversely proportional to the elastance. So at smaller alveoli and the airways, it is the compliance or the capacitance which plays a major role in actually determining the reactance, because the inertance is vanishingly small over there, almost nearing to zero. Not zero, nearing to zero. So this is how the reactance is. Reactance is actually, omega, as I said, is always a function of the frequency 2 pi f. Its inertance, that is I, minus, that is by convention, 1 divided by, the denominator comes the capacitance because it's the elastance. So this is how the formula of the reactance is. So what happens? If the capacitance decreases, then the entire parameter 1 divided by omega c will increase. Increase means become more negative. If the capacitance increases, 1 divided by omega c, that factor will decrease. Decrease means it will become less negative or maybe more positive. So that means the reactance is a function of the inertance, and reactance is also a function of the capacitance. Now, why function of the capacitance? I have said, why haven't I put it reactance is a function of 1 divided by capacitance? Because it is always a function which is after I minus something. So that means if this is 1 by omega c, it becomes more negative. Okay, that means the I is vanishingly small, it's nearing zero, but it's not zero. Then what happens is a 1 minus 1 by omega c, it increases. So if x increases, it increases. So this is just for the sake of understanding about this, because we do not need this application that much, but you need to understand that what is actually behind it. Okay, so this is how this formula goes about the capacitance. So before I move forward, means, is there any doubt or something is not clear? Or Salvi, if you like to add something, Disha would add something, or anybody need to ask anything at this juncture? Over here, right.
Interpretation will cover all the other the directions.
Left, the resonant frequency and
Okay,
X,
okay,
that is I'll just cover. So, okay. So, see, now if you are plotting this the reactance curve. On the x-axis, there are the frequencies, and the y-axis, there's a reactance. Now, as I have said, that the reactance is actually inertance minus the capacitance. So if the reactance curve is plotted from a very low frequency as we go up, so that means as we are moving up, the inertia starts increasing. As I said, initially it's vanishingly small, but then it starts acquiring some numbers, though albeit small, but start acquiring some numbers. So maybe at a particular point, what happens is that the inertance and the capacitance becomes numerically equal. But since by convention they're opposite, 1 minus C, that means the entire reactance becomes 0. So that frequency of the sound wave, where the entire reactance of the system is 0, it is called a resonant frequency. And at that particular frequency, what happens is that independence is given only by the resistance, because X has become 0. So this is what is called a resonant frequency. Important application of resonant frequency is that this metric changes in disease state. Okay, so that is the importance of it. Now, if you again look at this same plot, so if you see that there is a yellow triangle, so that is basically all the integrated reactances from the lowest frequency till the resonant frequency. So that plot is an integrated plot. It's an area or the integrated area. So that is what is known as the integrated reactance, or AX. This triangle is also called Goldman's triangle. Now this AX is a parameter, and all these parameters, so whether it is a FRES or AX, these all are the parameters. See, they are given the parameters which is telling us about the smaller airways, because we can understand that these are for the sound waves which only reach the smaller airways and comes back, because the high frequency sound waves do not reach there. That means we have the resistance parameters, resistance at 5 Hz, resistance at 20 Hz, resistance R5 minus R20. Then we have reactance parameters. Reactance, since we are interested in the smaller airways, so we take a reactance at 5 Hz only. And we have this FRE, that's a resonant frequency. And we have this integrated reactance, or the AX. And this AX has been shown to be the most sensitive parameter to identify disease state. Now, what the disease state is, that we'll see, but it is very, very sensitive. May not be specific, because by being very sensitive, it loses its specificity. So that is a word of caution, but it is very sensitive. So I think this is the reactance curve. This is usually a slope. This is not a straight line, because reactance is always a function of the frequency of the sound waves. And 0 is the resonant frequency, and this is actually the normal curve that we get. So this we have talked, resonant frequency, I just talked about it, normal 6 to 12 Hz. This is the reactance area, or the AX, or the integrated reactance.
Disha, just 2-3 slides visually. I think you'll do it post lunch. Okay. So thank you, Dr. Moitra. I think very exhaustive and complete presentation of the principles. So I'm just going to summarize 3 points so that you all can start digesting this before you start digesting the food. And once you have digested the food, we'll come back again, and then we'll revise it a little bit and then go through the interpretation after the hands-on and the demonstration. Okay? Simple things. In oscillometry, we're using an oscillating pressure wave. A pressure wave, okay? A pressure wave which is oscillating at different frequencies. Now, this pressure wave can be generated by 2 or 3 different ways. One is by sound. Sound is a pressure wave. Second is an oscillating piston. That is also generates pressure waves. And the third is, what is this use here? A vibrating mesh. A vibrating mesh also generates oscillating pressure waves. So 3 different ways in which you can generate oscillating pressure waves. Now these oscillating pressure waves are sent at different frequencies. Sir, do you listen to radio? AM radio and FM radio. Which one do you like? You like FM. Why do you like FM? The correct. AM uses low frequency. But what is the advantage of that AM? You can play it all over the country. Weak, it may not give you very good quality sound, but it travels across the length and the breadth of the country. All India radio. FM is high frequency, travels only shorter distances. So only in Kolkata. Radio Mirchi. The same Radio Mirchi will not go anywhere else, right? Now, using that principle, Dr. Moitra very beautifully said, low frequency travels long distances, so low frequency will travel across the entire respiratory tract. High frequency travels only up to the larger airways, right? So 20 hertz will travel only in the larger airways, 5 hertz will travel right up to the end. And when you subtract R5 minus R20, what do you get? Small airways. That's the beauty of this test. You will get information related to the small airways. Fundamental principles of sound waves. And the parameters that we get, resistance and reactance. Reactance is a combination of inertance and capacitance. We will revise that again after lunch. Okay? So, yes. Please. What about? Good. We'll cover that when Dr. Disha talks about how to perform the test. She'll tell you everything about what are the variables that will affect the test. Your tongue is there, your cheeks are there, your body position is also there. You know, all these are very important determinants of your test. So that will be covered in the post-lunch session. That will also have an impact. Yes, you want to answer that?
Yeah, very right. See, it all depends upon that what instrument you're using, what sound wave frequency you're using to detect what. So that means to pick up the factors of the chest wall, you need to have a sound wave which can reach the chest wall and come back. Okay. If you're using a sound wave which cannot reach the chest wall, but only reach till the periphery of the lungs, it will give you only information about what is there till the periphery of the lung. Usually for majority of the clinical purposes, what we're using the resistance at 5 hertz, okay, it cannot traverse the entire chest wall. You need much lower frequencies, say 3 hertz, okay, which can traverse the entire chest wall and come back. So that's why we said that if you want to know about the chest wall, you need to throw in a lower frequency sound wave, which can go up to that level and come back. So that is how you just choose your frequencies and you just use it. Say if you want to just using a microscope and you want to see that you change the objective, no? So that's how 5x, 10x, 20x. It's similarly here also. It is called a frequency dissection of the entire system. You just use different frequencies to look at different distances and pick up the actual information for the different levels. That's how you do it.
So should we break for lunch? Yeah. We'll come back in how many minutes? 40 minutes?
How to perform. We have our lungs perform two very important functions. One is ventilation, second is perfusion. We all know that. One is taking in air from the environment into the lungs. Second is letting that air, the oxygen in that air, dissolve with the blood. That is called as perfusion. So these are the two important functions that your lung performs. Okay? Why is this important? Because our body requires oxygen to nourish itself. And in order to utilize this oxygen adequately, you need patent airways. Okay? Now, we have majority of the people in our country having obstruction in the airways, which is undetected, undiagnosed. So how do we do that? We have a very important tool, that is the spirometer. Spirometer is the gold standard. Like both the speakers in the past have mentioned, the spirometer comes with a lot of challenges or a lot of limitations.
I will not go into detail, but these are the challenges that the spirometer offers, though it is the gold standard. On the other hand, we have the lung oscillometer. The lung oscillometer is a highly sensitive tool which helps you pick up small airways obstruction, very sensitive in comparison to the spirometer. However, the only limitation that we see right now with the lung oscillometer is we do not have the cutoff values, or we don't have the predicted equations for the Indian population. But having said that, it is a highly sensitive tool and it is the future of lung function testing. This list has been shown by Dr. Sandeep Salvi on what are the applications of lung oscillometry. I will not get into the details of this, but I would just like to take you all through the principle. Lung oscillometry in comparison to spirometry is a very simple test to perform. It can be performed in children, it can be performed in the elderly. All those of your patients who are unable to do spirometry, lung oscillometry is the end solution to it. Now, what is the principle that is used in lung oscillometry? Very simple. You see the cartoon over here? Okay. Consider this as your circuit. Okay, in physics, a circuit. Current is flowing through the circuit. Now this is the flow, this is your current. When you offer resistance or when you offer an obstruction, what is going to happen to the flow of the current? The flow of the current is going to reduce and the pressure is going to increase. This is the simple principle that is involved in your lung oscillometer. Increase in resistance, your pressure is going to reduce and your pressure is going to increase and your flow is going to reduce. Another example is a pipe flowing with water. You all are watering plants in your garden. At one end you have the tap, on the other end you are watering the plants. Somebody comes and stamps on the pipe. What is going to happen to the flow at your end? It's going to reduce, but the pressure on the tap is going to increase. This is the principle used in lung oscillometry. What is lung oscillometry? If you remember, sir mentioning there's something called as impedance. What is impedance? What is lung oscillometry? This is a person breathing normally in and out. Lung oscillometry involves normal tidal breathing, just like how you and me are breathing right now. When you pass oscillatory pressure waves, remember in lung oscillometry, changes in the pressure and changes in the flow are measured. So when you pass in oscillatory pressure waves, remember sound waves, as mentioned by sir, sound waves are pressure waves. Sound waves are being passed into your respiratory system. These sound waves go and superimpose on your normal tidal breathing. Depending on the obstruction in your airways, there is going to be a change in the pressure and a change in the flow. Remember the principle of lung oscillometry. Now that change in the pressure and the flow together, because of the resistance, because of the obstruction in the airways, is called as impedance. Impedance is denoted as capital Z. Okay? So as of now, just remember impedance. Impedance is any kind of obstruction in your airways, all of that together is called as impedance. These oscillatory pressure waves can be generated by two different mechanisms. One is the sound, second is the oscillating piston. Now there's a very unique property of this sound. Sound waves or pressure waves are passed into the respiratory system. Now, Dr. Salvi gave a very interesting example of the radio Mirchi. We all listen to the All India radio, the Akashvani. The same radio jockey is being heard across the country. Okay? Whereas on the radio Mirchi, which is very localized to Kolkata, you will not have the same radio jockey speaking the same content in Kolkata versus the person speaking in Pune. It's going to be different. Now, why is this so? because of this very unique property of the sound. Now, what is this unique property of the sound? Sound waves of a higher frequency. Now, the All India radio, the sound frequency is very small. It is only 1 megahertz. Hertz is the unit of sound. So the AM that you listen is made up of a smaller frequency. So smaller frequency sound waves travel longer distance. That is why you are able to hear the same radio jockey across the country versus the Radio Mirchi. The Radio Mirchi uses a higher sound frequency of 100 megahertz, whereas your AM uses 1 megahertz. So therefore, your Radio Mirchi is heard only in your area. So sound waves of a higher frequency travel shorter distance, and sound waves of a lower frequency travel longer distance. Let me give you another example. Any music lovers over here? I'm sure in Kolkata there are a lot of music lovers. Now, if you are sitting in an auditorium listening to music, you have the sound notes sa, re, ga, ma, pa, dha, ni, sa. Okay? The sound note sa and the sound note ni. I just went on to Google and checked the frequency of these sound waves. Now, sound wave sa is 256 hertz. Sound wave ni is 480 hertz. What do you think? Which sound wave, which note is going to be heard more clearly to the person sitting on the last bench? Sa. Why? Because it is of a lesser frequency. So a lesser frequency sound wave is going to go farther as compared to a higher frequency sound wave. Now this is applicable in your lung oscillometry. We know sound frequencies are passed into the respiratory system. Sound frequencies generally ranging from 5 hertz to 30 hertz is passed into the respiratory system. The lungs, you have the airways into 23 generations. From the 1st to the 7th generation is your large airways. From your 8th to the 23rd generation are your small airways. Now remember, in lung oscillometry, pressure waves are sent in on your normal tidal breathing. If I pass a sound wave of 20 hertz, it is only going to travel up to your large airways because 20 hertz is a higher frequency. So it is going to measure resistance only up to your 7th generation, that is your large airways. Now if I pass a sound frequency of a smaller sound frequency of 5 hertz, this is going to travel in your entire respiratory system. Why? Because it is of a lesser frequency. So a sound wave of 20 hertz, your lung oscillometry will pass sound waves of 20 hertz, that will give you resistance in your large airways. When your lung oscillometer will pass a sound frequency of 5 hertz, this will give you resistance in your total airways, right up to the small airways. Why not less than 5 hertz? Because when sound frequencies are passed, you all will be doing the test, you all will notice that there are vibrations that are happening. Okay, now these vibrations should not get distorted before they reach up to your small airways. Therefore, anything less than 5 hertz is not taken. Why not more than 30 hertz? Is because it causes discomfort to the patient. So in lung oscillometry, you use sound frequencies ranging from 5 hertz to 30 hertz. So these are sound waves being passed. Now sound waves, like sir mentioned, can be of different frequencies. You have mono frequencies, you have multiple frequencies. So, and all sound waves are post oscillations. Okay? So you have the single frequency sound waves called as mono frequency, very rightly mentioned by sir. So first you have the 5 hertz going in, followed by 12 hertz, then followed by 20 hertz. So one after the other is called as mono frequency. What is the IOS? You all must have heard of the IOS, that is the impulse oscillometer. What happens in the IOS? All these frequencies are clubbed together as an impulse and then sent into the respiratory system. There is a third type called as the pseudo-random noise. So these are the different types of sound waves or the frequencies that are passed into the respiratory system. It does not make a difference as to which sound frequency is going in, whether it is mono or whether it is an impulse. What are the different parameters that we measure in lung oscillometry? Like sir mentioned, you measure the impedance. Now what is impedance? Impedance is all the resistance, all the obstruction in your airways together is called as impedance, denoted as capital Z. Now, impedance is composed of two important parameters. One is the resistance, second is the reactance. So, resistance plus reactance will give you impedance. What is resistance? Resistance is offered by the airways, by the chest wall. Anything that is opposing the pressure waves that you're sending into the respiratory system is called as resistance. We all know what resistance is. Resistance is denoted as capital R. Now, if I want to measure resistance in the large airways, that parameter is going to be called as R20, because 20 hertz is going to give me resistance up to the large airways. They are not going to go into the small airways. So resistance in the large airways is denoted as R20. Resistance in the small airways means 5 hertz. Smaller frequency will go up to your small airways. Resistance in the total respiratory system is going to be denoted as R5. Now, if I want to know the resistance in the smaller airways, I'm going to subtract R20 from R5 from R20. So R5 minus R20 will give me resistance in the small airways. So your lung oscillometry report will have R5 resistance in the total respiratory system, R20 resistance up to your large airways, R5 minus R20 resistance in the small airways. There is another very important parameter that is measured in your by your lung oscillometer is reactance. Now, reactance is comprised of inertance plus capacitance. Our lungs are a very elastic organ. They extend and they come back to their original position while breathing. Okay? Now, what is reactance? Reactance is denoted as capital X. Reactance is formed by two components, inertance and capacitance. If you remember Dr. Salvi's presentation, he told you something about the functional residual capacity, or the FRC. Your FRC is the volume of air that always remains in your lungs and never comes out, no matter what you do. Now, this air that is already present in the lungs is offering some kind of resistance when you send in pressure waves. That resistance is called as your inertance. What is capacitance? Your air sac or the alveoli are very elastic in nature. They extend and they come back to its original position. That elastic property of the alveoli is called as your capacitance. Capacitance is dominated in the small airways. Okay, so I hope these parameters are clear. What resistance is, what reactance is. Reactance is a combination of inertance and capacitance. Inertance is the resistance offered by the air that is already present in the airways, and you have the capacitance is the elastic property of the alveoli. Again, your capacitance is of two components. One is compliance, other is elastance. So like Dr. Salvi mentioned, compliance is how much it can extend. Elastance is how fast it comes back to its original position. So that it depends on different lung conditions, depending on the thickness of the alveoli. So once again, to go in a little bit of detail, this is your resistance. That is, your resistance is offered by the chest wall. All the arrows that you see are the resistance that are offered to the flow of air, to the flow of sound waves. That is resistance denoted as capital R. Then you have your reactance. Reactance, like I mentioned, is inertance and capacitance. Inertance is the residual volume of air that is offering some kind of resistance to the flow of air. That is called as your inertance. Your capacitance is the elastic property of the alveoli. Okay? Again, your capacitance is dominated in the small airways. Capacitance is formed of two components, that is your elastance and your compliance. To sum it all up, what are the different parameters that are measured in a lung oscillometry report? So you have resistance at 5 hertz, resistance at 10 hertz, depending on the sound frequency that is passed in. You have reactance. Reactance is denoted as capital X. So reactance is also measured at 5 hertz, 10, 15, 20. Small airways resistance is R5 minus R20. Resonant frequency is denoted as FRES. What is resonant frequency? We will do that. And you have your area of reactance denoted as capital AX. I think, sir, you'll take this. Okay, so this was all about the basics of lung oscillometry, which I thought I would sum up from Sir's talk and Dr. Salvi's talk. Now we will move on to how to perform lung oscillometry. Like I mentioned, it's a very simple test to perform. Like spirometry, if you all have done spirometry, you all know that your patient has to put in a forceful maneuver, has to forcefully blow into the device. If he or she does not, you may end up getting a wrong report. On the other hand, your lung oscillometry is a very simple test to perform. Why? Is because it involves normal tidal breathing. These are the technical standards for respiratory oscillometry, which the ERS has released in the year 2019. I suggest you all read this to get into the details of what is what of lung oscillometry. Okay. Now, these are some of the prerequisites that you all need to take care when you all are performing your lung oscillometry test. Lung oscillometry test is generally done in normal tidal breathing in and breathing out normally. You need a trained staff for this particular test. I know it's a simple test to perform. However, it's very important because this test has never been done by any of your patients, never seen. So it is very important for your staff to be trained or you to be trained before getting this test done. Patients should be breathing in a relaxed and a stable manner, should be seated in an upright posture with the correct head position, not bend downwards or too high. It should be in the right level. Sitting position is preferred with uncrossed legs to reduce extra thoracic pressure. Cheek support is very important. Something like this. Why? Is because if you do not offer support to your cheek, there is some additional resistance that could be added to your output, the result that you get. So you need to have a cheek support. Mouth seal. The mouthpiece should be held tightly with the teeth, sealed with the lips. There shouldn't be any air leak happening from the sides of the mouth. Tongue position. The tongue should not obstruct the mouthpiece when the patient is blowing in and out. Prior to measurement, a visual check is very important to observe your patient visually, whether he or she is performing the test correctly. Use of a nose clip. We don't want to measure the air that is being taken in through the nose. Therefore, we put a nose clip. Very important that the patient breathes in through the mouth and not through the nose. Tidal breathing, depending on the type of device and the type of frequency that you all have opted for, the test timing will be determined. However, the normal range, every trial lasts for 30 to 40 seconds. A minimum of 3 readings should be taken pre. If you're doing a post bronchodilator, you do another post 3 trials. Steps while performing the oscillometry. Explain to the patients what this test is all about. Why you are doing this test? Because they know what a blood pressure test is. They know what an ECG test is. They don't know what a lung oscillometry test is. They've never seen it before. So explain to them. Explain to them that you all are going to experience vibrations. The vibration is going to increase after a few seconds. So explain to them all these points. Demonstrate the test to them. Have a mouthpiece with you. Show them how the test is done. Show them how to hold the mouthpiece. Show them how breathing is done. You remember, you need to do normal stable breathing, not too high, not too low. This may end up giving you wrong results. Head and body posture. Visually, just look at your patient whether he or she is sitting correctly, sitting properly in the with the right head position. And finally, start the test. Here is a small video on, we've just a very preliminary.
Before you perform the lung oscillometry test, it's very important to explain to your patient
If we can just have increased volume of this.
The prerequisites required before doing the lung oscillatory.
Okay, till they do that, I just want you all to take. I just want to take your attention to what is the correct technique. Nose clip is very important. Cheek support is very important. Subject should be breathing in a normal, stable state, not too high, not too low. Seated in the upright posture, mouthpiece should be correctly held, and you should have the correct tongue position. That is, the tongue should not obstruct the mouthpiece. Okay? What you see over here is a test done on a mono frequency. That is, one sound wave is being passed at one given point of time. The above graph is the flow, the below is the pressure. Remember, in lung oscillometry, you are measuring changes in the pressure and the flow. You need to have uniform flow. It should not go below the normal, and it should not go above the standard. So these waves that you see, it's a 23-second test. Okay? So this is the 5 Hertz followed by the 12 Hertz. So if you notice, you can see the differences in the pattern of sound waves. This is 5 Hertz, this is 12 Hertz, and then this is 20 Hertz. So this is the time, the clock or the timer. So this particular test is for 36 seconds. This is how a good lung oscillometry test should be done. However, when we will actually do it, demonstrate it, it will be much easier for you all to observe. So this was all about how we perform a good quality lung oscillometry test. Now what we will do is, we will have a volunteer from your end. We have somebody from the, and when we do the hands-on, we have a couple of devices. We will divide ourselves into 3 groups. So we have the iOS, which is an impulse. We send an impulse into the respiratory system. We also have another device, which is which is a pseudo tremor flow. And then we have the Antlia, which is a monofrequency device. So now we would demonstrate it on the Antlia, which is a monofrequency. So if I can have any volunteer who will come and, oh please, Mr. Kamath. Yeah, I'll do that. So this is the software of the Antlia device, which is already present in my system. The first thing that I would do is fill in the details of the patient. So the device is connected to my system. Okay, I'm going to put in the patient information over here. Your last name? CHOW? AHURY. Age is 44. Height. Please do not ask your patients a direct height. We advise you to actually measure it before getting the test done. Weight. Smoking status. Which part of the country do you belong to? Okay, now the patient's details have been saved. We are now going to instruct the patient on how to perform the test. It's a pre-test, that is this pre-test as in pre-bronchodilator, frequency mode. Now this particular device offers you different frequencies. You can select any single frequency and start the test. If you want to know resistance in the total airways, you can just select mono 5. Okay, if you want to know only up to your large airways, you would select mono 20. But here I want to understand the complete status of the lungs, so I'm going to select on multi-mode frequency. What is multi-mode frequency? on its own, automatically the device is going to first send in 5 hertz, then it will send in 12 hertz and then 20 hertz. I will not have to manually select the frequencies. So selected multi. Okay. Now if you notice the volunteer over here, there is a mouthpiece that is attached to the device. You need to hold your cheek slightly. Ensure that there is no air leak happening from the side of your mouth. Okay, hold the mouthpiece tightly with your teeth, seal it with your lips. You don't have to breathe through the nose. When you are breathing in and out, normally just like how you are breathing right now. Okay, you should not breathe very high, or should not be too fast. You should not breathe in too slow. Okay. Now we will put on the nose clip once you start breathing. Now as we start breathing, this test will last for around 36 seconds. After every 12 seconds, the frequency is going to increase. So the vibrations are also going to increase. So you don't need to get scared. You just have to hold it or hold the thing tightly and just keep breathing normally until I tell you to stop the test. So I've started the test. The device gives you a few seconds to get used to the normal tidal breathing. So this is just the initial phase before the patient starts breathing in and out. I don't know if you are able to hear the vibrations. Inhale and exhale. We have seen
This is 5 hertz. It is 5 hertz.
This is 5 hertz. Now you will automatically see the difference between 5 hertz and 12 hertz. They are much closer to each other. 12 hertz is a higher frequency sound wave. This is your 20 hertz. Much more closer, much more compact. So if you see, he kind of took a slightly higher breath in between. So the first trial is done. We will do 2 more trials. You need to have 3 free. It's okay. Yeah. Start. Breathe in and out normally. So this is the 5 hertz that is going in, followed by 12 hertz. The above graph that you see is the volume of air that is being blown into the device. Then you have your 20 hertz. Okay. We'll do one more last trial, then we'll do only 2. Okay. Okay, so what you see over here is the first trial, that is Act 1, and the second trial, that is Act 2. So what is the report giving us? It is giving us resistance at 5 hertz, the first parameter R5. It is giving us resistance at 12 hertz, resistance at 20 hertz, R5 minus R20. It also gives us reactance at 5, 12, 20 hertz, and then it also gives us impedance, that is capital Z. Now there's something called as, in spirometry, you have a couple of pointers to identify whether the particular trial has been done correctly, whether it's a good quality trial or not. In lung oscillometry, there is nothing like this because the patient is breathing in and out normally. But believe me, even with normal tidal breathing, there are errors that can happen, like a glottic closure. The mouthpiece can be obstructed because of the tongue. Maybe the patient stops breathing in, is not breathing out, there is air leak that is happening. So these all errors are possible in lung oscillometry. However, they have not been documented yet, not even in the technical standards have documented the quality issues in terms of lung oscillometry. However, there is something called as the COV value, covariance. Covariance tells you how much of variation is present in all the given trials that the patient has performed. Now here there are two trials that we've done. This particular software will give you the COV value of the average of 3 trials. So only after you perform 3 trials, you will have an average. That average will decide your COV value. So if your COV value is more than 10%, anything less than equal to 10% is acceptable. Anything more than 10% is a poor quality lung oscillometry test. For adults. For children, it is 15%. However, this is just mentioned in the guidelines. There is a lot to be explored even in this area of quality assurance. So this is just for your information. So I would now end my talk over here. If you have any questions, we can take it now, or we can also do it during the hands-on session that we will have post the case studies and the interpretation talk. So, we can have the interpretation of lung oscillometry now.
Okay. Yeah, so thank you and once again welcome post lunch. I believe this is going to be an important talk. So keep your eyes and your mind open because interpreting oscillometry reports is not mentioned anywhere very easily in any book. So this is through experience, through a lot of reading, that I'm going to share some basic concepts about interpretation, and I'll show you some case reports as well, which I believe will give you the real insight into interpretation of oscillometry. Now, let me start by showing you this. Spirometry, when it started, one of the challenges was, do we have good predicted values for spirometry? Why did that concept of predicted values for spirometry start? That is because your spirometric parameters change between gender, males and females have different values, and it changes dramatically with age. So here are the normal spirometry values in different ethnic regions. So from the age of when you're born till the age of 20, your predicted FEV1, FVC values, they keep on increasing. They reach a peak by around 20, and after 20 there is a slow and steady decline till the age till you die. What does it tell you? That at any given point of time during your age, the values are different. And how much do these values contribute to the predicted value? Well, age, gender, height, weight, ethnicity, they all contribute to 80 to 90% of the variability. In the sense, if you have to interpret your spirometry report, you have to have correct values for each of these. And then you'll get a reference value or a predicted value, and then you compare your patient's value with the predicted value. And then you express it as percentage predicted. As simple as that. Okay. So for all these years, we have been using predicted equations for spirometry. And with this background, we also believe that even oscillometry will have predicted values. So oscillometry values will change with gender, it will change with age, it will change with height, it will change with weight, and it will change with ethnicity. That was a belief. My PhD student, Nitin Vanzare, He did his PhD actually on this, trying to derive the predicted values for impulse oscillometry for the Indian population. So Indian population, he said he did it only in Pune. That poor fellow spent 5 years taking his portable iOS to temples, to bus stations, to gardens, to community. It had to be a community-based study. It cannot be a study that you can do in a clinic and call all healthy people. So it had to be a realistic study. So he transported this 25 kg suitcase wherever he went. And for every 10 people, he got one healthy individual, for every 10 people that he screened. So it was a very, very hard work that he did for 5 years with the hope that he'll find a predicted equation for oscillometry. 1,200 healthy individuals were studied. Now, what he found was something very depressing, that all the traditional parameters, age, gender, height, weight, they contribute to only 10 to 20% of the variability. And here are the results. This is in contrast to what we normally find in spirometry. So for spirometry, it is imperative to have predicted values. Whereas for oscillometry, well, these values contribute to only 10 to 20%. There is no good predicted equation. That fellow went into depression, poor fellow. Said, sir, I'm writing this thesis and I'm not getting a good quality equation. Nothing getting more than 15 to 20%. Then I said, what is the review of the literature? And he said, sir, review of the literature also shows the same thing. Whether the study was done in Kolkata as well. I mean, you're the one who derived the predicted equation, one of the first predicted equations for the Indian population. Kolkata, Pune, Australia, Germany, generally you get a straight line, a flat line. There's no nice slope line to show that there is a good variability with all these parameters. Now, how do you then take the field forward? No good quality predicted equation. How do you interpret oscillometry? And then suddenly, sometimes you get that dimag ki batti, tuck, suddenly that light switches on. I said, maybe this is a good thing. That it doesn't have a big impact on your predicted values. Something like your blood pressure. What is normal blood pressure? 120, 130 by 80 or 80 to 90. Irrespective of what your gender is, irrespective of what your age is, irrespective of what your height, weight, whatever. So standard value, one single cutoff value. The same is true for diabetes as well, blood sugar levels. Your blood sugar levels don't vary with your age. Your blood sugar levels don't vary with your height. There's a single cutoff value for the blood sugar as well. So for many parameters, there are single cutoff values. And then we said, maybe perhaps there is a single cutoff value for oscillometry. We don't have to break our heads to determine the predicted equation or the predicted values and then interpret our oscillometry findings with reference to the predicted value. Maybe there's just a single cutoff value. And when I was listening to some of these presentations, Professor Brian Lipworth, who is from University of Dundee in the UK, he was mentioning about all the studies that he did. And he said, I used a baseline cutoff value of R5 of this much to be included in my study. And then I asked him a question, but then, so what percentage predicted were the R5 values for your asthmatic subjects whom you treated with so and so, and then you examined them using oscillometry? He said, no, Sandeep, it's very difficult to get predicted values correct. I have used single cutoff values for my asthma cutoff, for my COPD cutoff, whatever. And then that actually struck me. So what I feel now, Dr. Mohitra, this is, sharing it for the first time, perhaps we don't need predicted equations for the oscillometry. We do, I'll tell you where we require it. But for an adult population, I believe there is a single cutoff value. And this is a preliminary report of, say, close to 100 patients of asthma, COPD, and healthy individuals that we ran through the ARISE network. We have a network of doctors across the country from where we collect all this information and make meaningful interpretation. So you can see that healthy asthma, COPD, we're getting very good signals in the sense there are nice cutoff values. Healthy is below this, asthma is above that, and so on. So based on that, we are just presenting this abstract at the forthcoming NAPCON to be held in November. And we believe that single cutoff values for oscillometry parameters are good enough. They give you reasonable sensitivity, reasonable specificity, and area under the curve. Some of the areas under the curves are pretty decent over here. So I think that was the first realization. This is not in the books yet, but this is through experience. So to interpret oscillometry, you don't need predicted equations. Let's start with that. Nitin also did a very preliminary analysis of healthy versus COPD. I mean, he had a database of all the patients that he was doing at CRF, and he said, "Sir, let me check if I'm getting a signal between healthy and COPD." And then again, you can see the area under the curve, and the AUCs are pretty strong, pretty robust. Very nice demarcation between healthy and COPD. So that is the realization. Now, with this background and some of the cutoff values that I have read from the literature, some of the cutoff values that we have obtained from the ARISE network, I believe this is an important slide for you. These are, I believe, let's call them as tentative cutoff values for all oscillometry indices, whichever machine you're using. So R5, R20, R5 minus R20, 4, 3, 1. Just remember that. 4, R5, 3, R20, and R5 minus R20, 4 minus 3 is 1. Simple, 4, 3, 1. For X, 5, it is minus 1. For AX, it is 4. And for FRS, it is 12. Now, these are tentative cutoff values. Let me put it that way. As more and more knowledge keeps on accumulating, as we keep on getting more and more data, I think these values will get on fine-tuned. But by and large, this is okay. Now, there are two units that are used in oscillometry parameters. Centimeters water per liter per second. So centimeters water is the pressure, and liters per second is the flow. Change in the pressure divided by the change in the flow is your resistance. As simple as that. So either you express your units as centimeters water per liter per second, or you express it as kPa, kilopascal units per liter per second. Some instruments give the units in kilopascal units, some instruments give it in centimeters of water. For all practical purposes and for the simplicity of the brain, centimeters of water, I think, is a easier parameter to get used to. So if you have kilopascal units in your machine, there's a very simple conversion factor for that, 10.197. You multiply the kPa by 10.197, you'll get centimeters of water. And then the 431 minus 4, 412 will hold true because these are in centimeters of water. So let us use this as a reference point for our interpretation, single cutoff values. Okay. Now, what I've been showing you over here is essentially only for the adult population, above the age of 18. Below the age of 18, it's a different ball game. Why is it a different ball game? Here is the answer. Look at the R5 values. So what is the R5 value that I said for an adult person is 4. 4 centimeters of water per liter per second, 4. For a child who is 3 years old, the now measured value is around 12. As the child grows to become an adult, it reduces from 12 to 10 to 7 to 4 till it comes to adulthood. So the R5 values, they reduce as the age increases in the pediatric population. It's important to know that, because if you apply the adult equation over here, then you'll say, oh, all these kids have got asthma, all these kids have got obstructive airway disease. Have you all understood this? Same for the X5 value. The normal X5 value in an adult is -1. So somebody who is 3 years old, a child who is 3 years old, the normal value for them is -5, and it keeps on getting less and less negative till you come to -1. And the same thing for resonant frequency. Very interesting. So for a child who is 3 years old, the resonant frequency is up to 30. And then as the age increases to adulthood, it comes to around 12 to 14. So that is normal resonant frequency for children. Now, why am I saying this over here? If you plan to do oscillometry in children less than 5 years of age, they have high resonant frequency, which is normal. So it's important to ensure that your device has got that breadth of doing frequencies. Some devices will stop only at 20. then you'll not get the values for children. You need to increase the frequency to 25, even perhaps even 30. So that's the learning from these slides. So by and large, if you look at the resistance, whether it be 5 hertz or 20 hertz, up to the adolescent age, the resistance values decrease. And after that, it's a straight line. So irrespective of what your age is, doesn't make much difference. Same for the reactance values. Early childhood, it is more negative. As you become an adult, up to the adolescence, the XY becomes more and more towards positive, and then you get a flat line. Have you understood this? Because this is the fundamental essence of interpretation. Without this, you will not be able to understand what it all means. So do other parameters not make any difference at all? What about weight? We know that weight is an important contributing factor because it shrinks the lung. In a way, it makes it a little more restrictive. So you get more small airways obstruction in obese individuals. So does that mean that oscillometry does not have any impact on weight? Well, it does. And there are some good studies more recently, as you can see here, 2015, 2018, that in normal individuals versus obese individuals. And this is effect of body weight on distal airway function and airway inflammation. You can see that obese individuals have got higher R5 values. Obese individuals have got higher R20 values. The X5 values, they don't change much. There's no significance over here. But the X20 values are significantly reduced in obese individuals. So this is the summary. There are several papers that seem to suggest this. So if you're doing oscillometry in a child and the child is obese, keep this at the back of your mind. Because an obese child by default will have these biased parameters. So what is oscillometry all about? You remember the flow volume curve in spirometry? Remember the volume time graph in spirometry? What is the equivalent of that in oscillometry? Here it is. Let me take you through this step by step. Here is the zero line. Okay, anything above that is positive, and everything that is resistance, respiratory resistance, is all positive. So respiratory resistance is on the higher side of the zero. Resistance is measured in centimeters water per liter per second. Below the zero line is your reactance value. Anything that is minus to start with at low frequencies is going to be your reactance. As your frequency increases, 5, 10, 15, 20, as your frequency increases, see what happens to the reactance line. The reactance line goes from negative to 0 to positive. Whereas the resistance line, whether it has 5 hertz, 10 hertz, 15 hertz, 20 hertz, it still remains a straight line. What is the message? In a normal, healthy individual, resistance is frequency independent. Doesn't care what the frequency is. It shows you the same value. R5 value must be the same as R20 value in a normal, healthy individual. Whereas reactance, it has got a negative component and then it has got a positive component. The negative component, which is the capacitance portion of it, is the one that is negative. So negative is the capacitance, and the positive portion above the zero is your inertance. So reactance, as I said, is made up of two components. Reactance is made up of two components, capacitance and inertance. Capacitance is the negative component, inertance is the positive component. The point at which the reactance line crosses the zero line, this point, this frequency at which it crosses, is called as the resonant frequency or FRES. Okay, it's called FRES or resonant frequency. Have you understood this? So what are the parameters that we evaluate in an oscillometry? From the resistance point of view, we have R5, we have R20, and then we have R5 minus R20. Simple. From the reactance point of view, we have X5, we have X20. By and large, we don't use X20 much. The only parameter we use is X5. And the area, the triangular area from 5 hertz frequency to the resonant frequency, this area is called as the area of reactance. called as the Goldman's triangle. And as Dr. Moitra said in the earlier session, most sensitive parameter for lung health, area of reactance. Area of reactance is critically dependent on what your X5 value is and what your resonant frequency is. So it depends on two parameters. That gives you the X5 value. So again, just to summarize, I hope you all are understanding, you're following whatever I'm saying. Resistance parameters, resonant frequency, reactance parameters. These are the traditional parameters that we see in an oscillometric report. However, there are additional parameters which I've started believing and understanding are equally important. What are these? The reactance at 5 Hertz during the inspiratory portion, reactance of 5 Hertz during the expiratory portion. X5 inspiratory, X5 expiratory. During your normal tidal breathing, you have inhalation, exhalation, inhalation, exhalation. During the inhalation, what was the reactance at 5 Hertz? During the exhalation, what was the reactance at 5 Hertz? I'll tell you the value of this. So X5 inspiratory, X5 expiratory. And then usually most of the instruments, they give you delta X5. What is delta X5? Inspiratory minus expiratory. They don't tell you the inspiratory value separately. They don't tell you the expiratory value separately. They just do the inspiratory minus expiratory, delta value, delta X5. Similarly, you can have X5 end of inspiratory, X5 end of expiratory. And also area of reactance, inspiratory, area of reactance, expiratory. But by and large, these are the parameters that you should be aware of. Does anybody need more clarification on this? I hope it's clear. Okay, can I take this forward? Okay, here it is. Pre and post bronchodilator. Can you see this blue dotted line? This is what a normal healthy individual's value should be. This dotted green line is the normal reactance of a healthy individual. This is the value of a patient pre-bronchodilator. This is post-bronchodilator. This is pre-bronchodilator area of reactance, sorry, X5 and the X20, and this dotted line is post-bronchodilator. Now let us try to interpret this. In this individual, what has happened to the R5 value compared to that of a normal healthy individual? What has happened? So overall resistance in the airways has increased, correct? What has happened to the R20 value compared to that of a normal healthy individual? What has happened to R20? Also increased. R5 is also increased, R20 is also increased. And R5 minus R20 is also, there's a nice big delta over here, isn't it? So R5 is increased, R20 is increased, R5 minus R20 is increased. The moment that happens, this is indicative of asthma. Okay? In an asthmatic individual, you get resistance increase in the small airways, you get resistance increase in the large and the medium sized airways also. After giving a bronchodilator, what has happened to the resistance at 5 Hertz? Gone down. After giving a bronchodilator, what has happened to the R20 value? Gone back to normal, isn't it? R5, it has not gone back to normal, but it has certainly gone down significantly. R20 is almost come back to normal. So how would you interpret this? Still overall resistance is high. More seems to be predominant in the small airways. Large airways have completely opened up. That's the way you would interpret this, right? In reactance, this light green is the predicted one. So there's a big drop in X5. So X5 has become more negative. What has happened to the resonant frequency? So this is the resonant frequency. This will be at some maybe 8 Hertz. What has happened to the resonant frequency? Shifted to the right. After giving a bronchodilator, you can see that the line has gone further up. So X5 has gone up over here, resonant frequency has come back over here, and the triangle which was this big has now become this big. This is a classical case of asthma. Okay? Have you all understood this? R5, R20, R5 minus R20, X5, AX. is all that you have to see. Now let me show you the COPD one. Normal predicted, well, straight line, normal predicted, so-called predicted. Look at what happened to the pre-R5 value in this patient and look at what happened to the pre-R20 value in this patient. What is classical over here? R5 is high, R20 is normal. So what does it mean? Predominantly small airways obstruction, predominantly. And there's a huge increase in R5. Compared to this increase in asthma, you will get a much bigger increase in R5 and a normal R20. After giving a bronchodilator, what has happened to the R5 value? Well, it has reduced, but not as much as what normally you would see in asthma. R20 is normal. So there is some bronchodilator reversibility. X5 in asthma becomes a lot more negative than what happens in asthma. The resonant frequency becomes more to the right. So for asthma, the resonant frequency is say about 15 over here. Here it is more than that. And then after giving a bronchodilator, you can see that there is a reasonably good improvement in that the triangle, which was this big, AX, has now become this big. So you get good bronchodilator reversibility signs on oscillometry. But that is all because of small airways opening up in COPD patients. Have you all understood the differences between asthma and COPD? Okay. The impulse oscillometry machine is here. And the impulse oscillometry gives a very nice figure. I like this figure that they put. So this is the image that they provide. On the left hand side of the image is all got to do with your resistance and the extrathoracic compliance. So let's focus on this first. This triangle over here, one triangle and another triangle, this depicts your large airways. So this is central resistance. This triangle over here, which is at the bottom, it depicts your peripheral airway resistance. So central airway resistance, peripheral airway resistance. And the bigger it becomes, the more is the resistance. So you can see that there is not much resistance increase in the central airways, slight increase in the resistance in the peripheral airways, assuming that this is the normal diameter. On the other side are the reactance parameters. What are the reactance parameters? You have the compliance, which is measured as elastance over here. So this is the compliance. The ability to expand. If this is your alveoli, how much it can expand is your compliance. And how much does it come back, how fast does it come back to normal is your elastance. So compliance is measured over here. Elastance is the speed with which it comes back to normal. So the bigger, it should be very small. Elastance should be just a thin line over here. So bigger the elastance, bigger the compliance, it means there is something wrong in the distal portion. And the inertance, which is a component of the reactance, is mainly in the large airways. So that's why it is shown over here. You can also measure the compliance of the extrathoracic large airways. So compliance of the upper portion, compliance of the lung, resistance, reactance that includes respiratory compliance and inertance. Have you all understood this? Because I find this graph, I find this figure very, very interesting. Now here is a case, which is a real case that we saw about just a little more than a month ago at a Pure Foundation. A 56-year-old male, she works in a school as an administrator, came to us because she had once come to Chest Research Foundation and thought that there are good doctors over here. She had asthma for many, many years, known asthmatic for about 20 years. But for the last 6 months, her asthma has been really getting bad. And she has needed to be on oral corticosteroids, 60 milligrams of prednisolone. And the moment you start bringing it down, her symptoms again start reappearing. So it has been a real challenge for her to get weaned off from oral corticosteroids. Now she went to a doctor who said, your Ig levels are high, asthma is not responding to your routine medication. I suggest that you start this injection called benralizumab. And so she was a little worried that, I have to take an injection worth 5 or 6 lakh rupees. Is it really worth it? I mean, am I so bad that I need to take an injection of this expense? So she came to us for an opinion. Now we did spirometry first. You can see the change in the FEV1, 18% to 170 ml. Well, reasonably good bronchodilator reversibility, isn't it? There's also some change in FVC, 10% 230 ml. The ratio was obviously showing obstructive airway disease, and she had significant, well, small airways obstruction as well. Now, this is the information that the spirometry provided to us. And then we said, why don't we do oscillometry? So we did impulse oscillometry in this patient. Now, this is the predicted resistance. So this is only the resistance portion. It gives you the resistance and the reactance in two separate graphs. So this is the normal resistance pattern, pre-bronchodilator and post-bronchodilator. What information is this giving us? 200 micrograms of salbutamol is producing a huge bronchodilator response, I thought. Look at the resistance at 5 Hertz. So this is kPa, this is about 0.9. So it's 9 centimeters of water. What is the normal range? 4. This is double of that. So the resistance is hugely increased at R5. Then there's a big drop in resistance, and well, it's high at R20 as well, but the gap between R5 and minus R20 is huge. So what is it telling me? She's got very, very high resistance in the small airways. When we looked at the reactance value, look at her reactance. What should be the normal reactance value? -1. Her value is more than -5. 5 times her reactance values have increased. Look at this, and the resonant frequency is close to 35. After giving us bronchodilator, it came down to -2. So -2, and as a resonant frequency came back to almost 18. Now, what this information gave me was, Arey, just 2 puffs of salbutamol can produce such wonderful effects. I don't think you should take benralizumab. What drug do you use? So she said, I use a Pulmocare device. Can you show us how to use it? And there's a fundamental error in the way she was using the dry powder inhaler. Now look at how the doctor who was treating her did not even check that. He just realized that she's a known chronic asthmatic or needs oral corticosteroids, and she needs to be put on benralizumab. These are the values. So I have converted all this to centimeters of water for simplification. And so from R5 has gone from 9 to 4.7. So here is my secret formula over here. 431 minus 1, 412. So R20, 3.97, come back to 3.26, not very far from normal, isn't it? R5 minus R20 is very high. It should be 1, it's 5. And after giving a bronchodilator, almost come back to normal. Look at the percentage change that this lady has had. -5 to -2. Area of reactance is 62, has come down to 11. And FRS from 35 to 90. Have you all been able to interpret this correctly now? Okay, can I go forward? I did something called as X5 inspiratory and X5 expiratory in this patient. Do you know what is expiratory flow limitation? What is expiratory flow limitation? These patients who have predominant small airways obstruction, their airflow closes during exhalation. So it's called as expiratory flow limitation. So your resistance, your reactance will increase during inhalation or exhalation? Exhalation. As you start exhaling, the reactance values during exhalation becomes more negative. -0.55, inspiratory is -0.35. This is kPa. What does it tell me? Expiratory flow limitation. Whenever exhalation reactance is more than inhalation reactance, there is some obstruction happening in the small airways. We treated this lady with Nivioli, the fine particle, ultrafine particle, beclomethasone with formoterol. And she says she's improving dramatically with that. That's the impact that this patient has actually taught us. Now here is another report. I want you all to interpret this now. I have put my so-called cutoff values over here. These are the figure. This is the tremoflow instrument. So I'm showing you results from Antlia, results from the Jaeger wire, and from Tremoflow. So what do you find over here? R5 is 9.9. What has happened to R5? Increase. What has happened to R20? Slightly increase. What has happened to R5 minus R20? Huge, isn't it? X5 become more negative. Area of reactance, which should be 4, has become 76. My God, huge. Resonant frequencies, 35. What does this patient have? Okay. Similar case. Well, so-called similar. R5 is 4.97. Is it raised? Slightly raised. R20 is 4.1. Slight. R5 minus R20. What has happened to R5 minus R20? Normal. Normal. Is there small airways obstruction here? No. You should say that confidently now. R5 minus R20, if it is normal, there is no small airways obstruction. As simple as that. X5 has become a little double negative. Area of reactance has increased. Resonant frequency has increased. Can you guess what this is? Yeah? You want to guess? Now I'm showing you something very important. X5 inspiratory minus 4.8, X5 expiratory minus 6.5. What does this confirm? So what is it? So it confirms the diagnosis of COPD. X5 inspiratory is minus 3.3, X5 expiratory is minus 1.28. This is? No, this is X5. So there is greater reactance during inspiration than during expiration. So this is restrictive lung, ILD, classical case of ILD. So this X5 inspiratory and X5 expiratory, I think I've learned a lesson, gives us tremendous vital information. Okay, have you all understood this? Now this has been published in the past, but it has been published in a very different manner. So this is characteristics of inspiratory and expiratory reactants in interstitial lung disease, published way back in 2013. In a normal healthy individual, this is delta X5, inspiratory minus expiratory. So in a normal healthy individual, which reactance is higher, inspiratory or expiratory? Inspiratory is higher, slightly. In an asthma patient, there is no difference between inspiratory and expiratory. In a COPD patient, what happens? Expiratory becomes bigger. Whereas in an ILD patient, it becomes higher than normal. Have you all understood this? This has been shown for the first time in 2013. But I did not understand the value of that until I saw these cases on my own. COPD versus bronchiectasis. There's a very recent paper that seems to suggest that if you look at the expiratory, so these are the 3 bars over here, healthy, bronchiectasis and COPD. This is the resistance. So they have shown whole breath, inspiratory, expiratory. Expiratory resistance, now this is R5, not X5. Expiratory resistance is significantly increased in COPD, whereas in bronchiectasis, it's very close to normal. So this is for resistance, this is for R5, this is for R20. Again, you see a big difference over here. R5 minus R20, much bigger difference. So COPD, bronchiectasis. In bronchiectasis, you also find some increase in R5 minus R20 and so on. So as more and more knowledge starts coming in, we'll learn a lot more. The interpretation of oscillometry is still evolving. What I've shown you is something that has evolved with time. There is no fixed textbook thing for that. Post-TB lung fibrosis. Can somebody interpret this for me now? Okay, let's look at R5. pre-bronchodilator, post-bronchodilator. So how much is the R5? 9. Is that normal? 4. So R5 value is increased. R20 value is also increased. So it should have been 3 over here, isn't it? So it's increased over here. R5 is increased, R20 is increased. After giving a bronchodilator, there seems to be a small drop in the resistance. But see what has happened to the reactance. -8. And the resonant frequency is around 35. Is there any change in the bronchodilator response? So what does this tell you? The severe fibrosis as you can see over here. So this knowledge is still evolving, and we will get better and better. Just to highlight some of the studies, just read this box over here. You don't have to look at the rest. Lung oscillometry indices predict COPD exacerbations in patients with group A and group B COPD. Now that is emerging knowledge. If you have your oscillometry indices, you can predict which patient is likely to get an exacerbation. Wow, that's good. That's good application. An area of reactance cutoff value of more than 0.5 kPa, so that's more than 5, was more sensitive, specific, and had higher positive predicted value and higher negative predicted value than FEV1. So even asthma can be detected earlier with oscillometry than with spirometry. I believe this is going to be the future. So you can see resistance values, reactance values, inhalation phase, exhalation phase. The moment you start seeing big differences between inhalation and exhalation, it starts giving you some more insights into what is happening inside your lung. So this intra-breath or within-breath oscillometry is going to be emerging as a very important diagnostic parameter. So the knowledge is still emerging over here. In Japan, they have an instrument called MedPro, and the software is so clever that they give you these colors. So this is how a healthy individual's oscillometry report looks like. This is what happens in asthma. This is what happens in COPD. Resistance values, reactance values. Healthy asthma, COPD. And the moment you see the images like these, then you don't even have to look at anything. Just look at the color, and the color will tell you, is this patient normal, abnormal, and what type of abnormality this is. Even the severity of airflow obstruction can now be categorized: mild, moderate, severe. So there are cutoff values that are now happening for R5. There are cutoff values that are now happening for X5. So severity of obstructive airway disease. So like how we have the spirometric indices for asthma, 60-80 rule. More than 80% predicted is mild, 60 to 80 is moderate, less than 60% is severe. For COPD, we have the 30-50-80 rule. So those rules are now slowly started coming for oscillometry parameters as well. Now I'm going to end with just showing you resistance values. Now here I'm going to show you percentage predicted. So this is only for the sake of simplicity, and let me see how much you all have understood. So who would like to interpret the report that I'm going to show? Would you like to interpret? Okay, here it is. So resistance at 5 Hertz, predicted is 0.26, so this is kPa. Observed is 0.4, which is 54%. R20 is also 54.5%. R5 minus R20 is normal. Is there obstruction? And if so, where is it? Is there obstruction? R5 value is increased, but R20 is also equally increased, and R5 minus R20 is normal. So where is the obstruction? Where? Excellent. Good. Pure proximal airflow obstruction. Now, would you like to do this? R5 0.26, 0.40, so R5 is increased. R20 is, well, close to normal. R5 minus R20 is 300% increase. Is there obstruction? If so, where is it? Is there obstruction? Yes. Excellent. Pure small airways obstruction. Okay. So one more. Would you like to do this, sir? Please. R5, 130% increase. R20, 81% increase. So 0.22 ka 0.40. And R5 minus R20 is 500% increased. Is there obstruction? Where is it? Huh? Excellent. Well done. Good. I'm going to stop over here. I hope I have been able to communicate the simplicity of interpretation, although it has taken us a lot of time to understand this. Interpretation of lung oscillometry and the secrets it is deciphering is still evolving and will get sharper and sharper as we get more and more data. And I think before we do the hands-on, Disha, quick 2 words on your network, because then I'll tell you what is happening. Yeah. Sorry? Please, please. No, let's do this first. Let's do this first. Yeah, yeah. So let me just put this into perspective. Lung oscillometry started in 1956, but it has taken a long time for it to evolve. And now we have reached a point of injunction where I believe oscillometry is going to go logarithmically upwards. So we are all who are sitting in this hall are lucky to be in the right place at the right time. This is the time onwards where oscillometry is going to become more and more popular. Now, in order to build the science behind it, we must contribute to the science generation as well. So, Disha will tell us about the network that we have established for lung oscillometry and what potential the network has to improve the science that we can decipher from this. So, Disha,
Thank you so much, sir, and thank you all of you all for your very kind attention. I'll quickly describe to you all what we are doing at our chest research and training in Pune. So we've set up this network called the International Network for Lung Oscillometry Research. It is one of the first network, I must say, in the world who is very specifically doing research related to lung oscillometry. As of now, so it's just 1 year old. And we have more than 30 doctors who are now registered and are a part of this network. We have Parthasati sir as well as Moitra sir also, who are a part of this network and contributing very actively. So we answer research questions related to lung oscillometry through this particular network. We also have a first abstract going for the NAPCON this year. So just in case any one of you all who are very passionate about research have access to the lung oscillometry device, I urge you all to please get in touch with me maybe after this workshop is over and we can talk over it. You all can be a part of this network, contribute and help. The first study that we are doing is deriving the cutoff values for the Indian population. You all can also contribute to it and be a part of this very much needed initiative. So thank you once again. And now I would like to invite sir to take ahead. Yes, some of the case study.
This is the experience of beginning. I do not understand. Dr. Salvi. Technician can do it. It's been sent across to Savala Center. I can get a report interpreted. It's a difficult business once I got into it. And last 10, 11 physics, mathematics, and reactants, reactants, etc. They're quite difficult for us to know at the age of 60 plus during the mathematics. So I'll be on it as a student. I'll project your knowledge and ask Charlie to interpret that. Yes, both of us. Oh yeah, both of you and also the audience. Let me see. A40. There's A40. Yes. Yeah. Have you got it? No, not this one. I have to go to a PowerPoint. Choose a PowerPoint. No, back, please. No, back again. Choose a PowerPoint. These are all folders. Go down. See the number of folders. Yeah. No, you have to just a minute, give it to me. I'll get them in classes. Sorted by types. I like a PowerPoint. PowerPoint. which wala? Could be. Last night only, last night, yes.
Individualize what does take out each factor and that's what we found while we were trying to do.
Very true. Yes, sir.
So I'll start from this. She's a 28-year-old lady. She had a tubercular infection and she was treated by me. And once she came this time, this was the result. You can see the resistance and reactance. I don't know how to interpret it.
So, Mr. Mandar, even if you can help. Now, what the anti device does is, depending on what your value is, it decides the scale of the y-axis. So, if you can see that this is resistance. This is the normal range up to 4.
Yeah.
So this green zone is normal. So at 5 hertz. Now, which one is pre and which one is post? So pre is this.
Post.
Pre and post. Going in the opposite direction, isn't it?
No, I repeated it twice. After this result came, I couldn't understand anything, head and tail of it. I said, okay, repeat it again. The same thing has come.
So even the post has gone in the wrong direction. So pre and post. So I believe this This could be because of maybe the mouthpiece is loose.
Probably.
Could
This will be always positive as you told independent typically.
be. Resistance can never go below, can never go in the negative direction.
So there is something wrong. This patient has a, because I asked, because this patient had endobronchial tuberculosis.
Oh.
Yeah.
Endobronchial tuberculosis.
Yeah, she had a definite endobronchial narrowing in multiple places. So I do not know if you look at the picture.
I would have expected the R20 to be increased.
Yes, but it is R14 or R12 is increased. So is it? No, it's on the reverse way. So anyway, I do not know the explanation of it.
I don't know what this means, really.
Okay. Well,
So we are still learning.
Anyway, let me go
So
to the next.
maybe Disha is an expert in understanding whether the test was done
Disha,
correctly.
can you enlighten us a little more? Okay. Acha.
The pressure and the flow waves.
Okay, it's not been projected here anyway, because I asked for some photographs. This is a young sporting person's calf. He had non-specific pain chest, and once it came to me, I said, okay, it seems everything is normal, go for a FOT.
Effort is also normal.
Absolutely normal.
Absolutely normal.
It's
So in the
supranormal.
well, it's in the range.
See the Goldman triangle hardly been anything.
Yeah.
So does it mean a good lung has got a smaller Goldman triangle?
The smaller the better.
Better the lung health,
Yes.
smaller is a Goldman triangle.
Absolutely. Yes.
So we can perhaps look at the athletes through this also.
Absolutely. Yes.
Yeah. So I projected it because he's a sportsman. So perhaps we can measure a sportsman
perfectly
lung
normal.
through this. Well,
Oh, this is interesting.
This
So
is-
pre is the red, the plain line. Post is this.
yes.
Pre and post. So this is very, very classical. Can anybody interpret this? Yes, ma'am. You want to interpret? Orange dress. Orange dress. Yes, ma'am. Come on. Get up. What has happened to R5? The plain line is the pre-bronchodilator. See, this is R5. What has happened to R5? How much has it increased? What is the number? No. It's close to 0.8, isn't it? It's above 0.6, so it will be 0.75 or something. What is the normal value? This is 7.5. So is it increased? Yes, R5 is increased.
So that is centimeter of water. You have to convert it to centimeter of water.
I've converted kPa to centimeters of water.
Achha.
This we're multiplying by 10. 10.179 or just roughly. What has happened to R20? So R20 is this one. So, is it normal or is it increased? So it should be 3 is normal, right? So it's close to normal. R5 is raised, R20 is normal. R5 minus R20, my God, there's a big gap. 0.75 to 0.3, 0.4. 0.4 means huge R5 minus R20. After giving a bronchodilator. See what has happened to X5? The blue line is pre-bronchodilator, dotted line is post-bronchodilator. So significantly reduced X5. Look at the area of reactants. My God, look at that triangle, is huge triangle. After giving a bronchodilator, the resonant frequency has come to 12 and the area of reactants has at least got better. So what does this tell you? Is there obstruction? Yes, no. Where is it? Entire respiratory tract or in the small airways? Only in the small airways. After giving a bronchodilator, what has happened? Significant improvement. So either this is SAD asthma or this is some element of COPD.
But why is a genu kind of thing?
What is the?
The pre-bronchodilator line, it's a genu kind of. See the resistance, there's a sharp angle, it's not a straight line. I mean,
This one.
I think it's because it is a FOT, it looks like that. Had it been a IUS, it would have been a curved line.
It would be a curve line. So I think they're developing that. Well, what is the diagnosis, sir?
Yeah, diagnosis, this is a FEV1 changing from 1.1 to 1.36. And FEF 2575.66 to 0.86. So there is a reversible airway disease, asthma. I couldn't understand the pre-bronchodilator reactance line going such acutely angled. So is it anything?
Yeah, so the post has gone down from -0.3 to -0.35. So 3 to 3.5 has gone down. Whereas resonant frequency is still. So what is? Reactance can increase during? Reversibility can increase reactance, is it?
Maybe in that case. This is a different patient. The diagnosis is BHR oblique asthma with allergic rhinitis. This is a patient's.
So there is R5 increase, R20 is normal, R5 minus R20 is quite big.
Yeah.
So there is small airways obstruction. There seems to be reasonable good bronchodilator reversibility. The X is not giving me what I should expect.
But the reactance hasn't changed much.
Yeah.
So there is an element of added element of interstitial, I mean stiffness to the
Possible,
lung.
possible. Did you find that?
No, actually, I'm looking for it. I have
Okay.
asked the patient for HRCT.
You agree with that, Saibal?
Saibal, anything? Absolutely. Yeah. What do you interpret from this curve? No, you have given this, we are looking at the time versus pressure, and is the shape of the curve has got anything to do to understand anything?
So how do you interpret this graph now? What is the graph telling us? So the logic, Dr. Parthasarathi, is you do normal tidal breathing.
Yeah.
Right? You need at least 3 tidal breathings per frequency for analysis. And I think what they're trying to see is, are you having at least regular 3 waves? Or is one wave like this, one wave like this, one wave like this? I think
this is checking the standard of the test kind of thing.
Yes, yes.
So if you get a long strip, it will be even better.
Yeah. So again over here, resistance is increased at 5 Hertz, normal at 20, slightly increased, I mean just 0.4. So it's slight reversibility, more towards normals, whereas the reactance is -3. So the only change that I'm seeing is reactance going from -3 to -2.5. So there is something happening in the distal portion of the lung. Now whether it is obstruction or whether it is restriction, we can't say that with this. We need to do that X5 expiratory.
Yeah, this is a case of interstitial lung disease.
Oh, excellent. Very, very classical, like similar to the one that I showed you, isn't it?
I think that's all. Thank you.
Good, please.
Yes, we have a reversibly defining criteria, but that's also evolving. Like we take 3 parameters, R5, X5, AX. Okay. X, we say if there is some says it's 80% change. 80% change in the AX, X5 50% change, and in R5 there's at least 30% change. So then we say it's irreversible. Okay, that's how. But there, if you look into the various references, they will just vary between. I was just looking at another reference, we are saying AX to be only 50%. Another reference saying AX 80%. These are the things. R5 around 30 to 40%, X5 50%, and AX around 80%. You check at the manuals that they give with the machine. Yes. Yes. Yes, the manuals they give with the machine, that you have to look into that, because various machines has different, and means that is what they have.
So I must compliment Dr. Parthasarthi for organizing this workshop. This is very visionary and futuristic. The belief that this is going to be a very novel learning experience for all the delegates. But what better than that, that there are 3 different devices sitting in the same hall for a hands-on session. I have not seen this in any meeting. So it's, I think, just shows the popularity that you have. But so we have the we have the impulse oscillometry, we have the tremor flow, and then we have the Antlia. So if you all can divide yourselves into 3 groups, make sure that you all go in equally into all these 3 places and do the test yourself and see how it feels. Sure. So any questions? Dr. Partha, any concluding remarks before we complete this?
You are the authority. I think this is my small comment. I think we need a long time to learn it. I'm at the very bottom of the learning curve, which is looking very interesting day by day. Once I started looking at it, it was quite difficult, kind of jargon, but now it seems getting clearer.
Good. I must thank Dr. Saibal Motra, Dr. Disha Ghorpade, and for giving such wonderful talks. All the 3 companies who have come here for the demonstration and hands-on. And finally, to each one of you all, it's so nice to see so many of you all sit in this hall right from the beginning till the end. Just goes to show that there is a lot of enthusiasm and interest in this. Yes, you want to say something?
So if the test report is we are making interpretation of small airway disease, how do we differentiate it's asthma, COPD, only based?
It's very difficult to differentiate between asthma and COPD. If there is only pure small airways obstruction, then you look at the whole picture. History, symptoms, clinical presentation, if required, any other diagnostic tool. Do spirometry. So I don't think we should stop doing spirometry. We should do spirometry and this as a complementary test. So look at the whole picture. And one big mistake I have done so many times in the past is just look at the report and make an interpretation. You may go wrong many times. Look at the whole picture. Look at the age, look at the gender, look at the symptoms, look at the X-ray pattern, look at everything, and then make a final diagnosis.
And we should not forget that for the adults, as in standing today, this oscillometry is still as a complement to spirometry. Okay, so it has replaced it. In children, it is done on its own. Children, we do not talk about the COPD. Okay, but in adults, definitely it says, as Dr. Salvia said, the history, patient's perspective, spirometry, and then we go on to do the oscillometry. So that's the entire thing. Yeah. No, see, there's nothing like that. Why? Because, as I've said, the resistance is always positive. Reactance is negative. It's a curve, it's a slope. And the resistance for all the normal matters, it's a straight line. So those are in a different curves. Okay. So reactance curves are different, resistance curves are different. Resistance curves move up or takes a U shape. Reactance curves moves to the right.
Good. Can you break up a bit? Please come here. Yeah. And please come here as well. Please do the test yourself. You have to realize how simple
Airway was. Airways. Okay.